Lesson 1. Introduction to Microbiology and Microscopy. Let us begin with the definition of our subject. Microbiology is the scientific study of microorganisms, those living things that are too small to be seen with the naked eye. It is a discipline that encompasses a vast diversity of life forms, including bacteria, viruses, fungi, protozoa, and microscopic algae. It may be difficult to comprehend, but the visible world, the trees, the animals, the people you see every day, makes up only a tiny fraction of the biomass on Earth. The true majority of life is microscopic. You are currently resting in a room that is teeming with invisible life, floating in the air, resting on surfaces, and thriving on your skin. Far from being something to fear, this is simply the nature of our reality. We live in a microbial world. To study this invisible universe, human beings had to invent a way to extend our senses. We had to invent a second eye, one capable of peering into the very fabric of life. This brings us to our primary tool, the microscope. Visualize a standard compound light microscope. It is a sturdy instrument, usually made of metal, sitting on a laboratory bench. At its base is a light source, a simple bulb. This light travels upward, passing through a condenser lens that focuses the beams into a sharp cone of light. This cone strikes the specimen, perhaps a thin slice of tissue or a drop of pond water on a glass slide. The light travels through the specimen and enters the objective lens, which performs the first stage of magnification. Finally, the light travels up the tube to the ocular lens, the eyepiece where you place your eyes, magnifying the image once more. But magnification alone is not enough. Imagine holding a magnifying glass up to a blurry photograph. You make the image larger, yes, but you also make the blur larger. You do not gain new information, you only gain size. In microscopy, the true limit of what we can see is not magnification, but resolution. Let that word settle in your mind. Resolution. Resolution is the ability of the lenses to distinguish two points as separate and distinct. Think of driving a car down a dark highway at night. In the distance, you see a car approaching. At first, it looks like a single, bright light. As it gets closer, that single blur splits into two distinct headlights. That moment, when the one becomes two, is the limit of resolution. Without good resolution, a bacterium is just a smudge. With good resolution, it is a defined shape with walls, internal structures, and purpose. The story of how we discovered this world is a story of curiosity. It begins in the mid-1600s. Picture a man named Robert Hooke. He was an English scientist with a keen interest in the new technology of lenses. In 1665, he placed a thin slice of cork under his crude microscope. Cork is the bark of a tree, dead and dry. Looking through the lens, Hooke saw a network of tiny, box-like compartments. They reminded him of the small, bare rooms that monks lived in, which were called cells. So, he coined the term cell. He was looking at the rigid cell walls of dead plant tissue, establishing the name for the fundamental unit of life. However, Hooke did not see living things. That discovery fell to a Dutch merchant named Antony van Leeuwenhoek. Leeuwenhoek was not a formally trained scientist. He was a draper, a man who sold cloth. But he was a master at grinding lenses. He built simple, handheld microscopes that were far more powerful than Hooke's. Imagine the scene. It is the late 17th century. Leeuwenhoek takes a drop of rainwater from a clay pot in his garden and places it behind his lens. He holds it up to the light. He expects to see nothing, perhaps just clear water or dust. Instead, he sees a chaotic, bustling city. He sees tiny creatures swimming, darting, and spinning. He called them animalcules, or little animals. He was the first human being in history to lay eyes on bacteria and protozoa. He looked at the plaque from his own teeth and found it teeming with life. He wrote letters to the Royal Society in London describing these wretched beasties opening the door to the microbial world. For a long time after Leeuwenhoek, scientists knew these tiny creatures existed, but they did not know where they came from. This led to one of the great debates in the history of science, the debate over spontaneous generation. For centuries, people believed that life could arise from non-living matter. They believed that if you left meat out, it created maggots. They believed that if you left grain in a dark corner, it created mice. They believed that bacteria simply appeared in broth, like clouds appearing in the sky. This idea is called spontaneous generation. It took a giant of science to disprove this and establish the foundation of modern microbiology. That man was Louis Pasteur. In the mid-1800s, Pasteur designed an elegant experiment to prove that life only comes from existing life. Visualize this experiment, for it is beautiful in its simplicity. Pasteur took glass flasks and filled them with a nutrient-rich broth. He then heated the necks of the flasks and bent them into a long shape, like the neck of a swan. He boiled the broth to kill any microbes already inside. This is the process of sterilization. Now, here is the genius of the design. The shaped neck allowed air to enter the flask because air is vital for life. But dust particles and microbes floating in the air would get trapped in the bottom curve of the swan neck. They could not travel up the steep slope to reach the broth. The result? The broth remained clear. It remained sterile. No life appeared, even though the flask was open to the air. The vital force did not create life. But if Pasteur tilted the flask so the broth washed into the neck and picked up the trapped dust, the broth quickly became cloudy with bacterial growth. This experiment proved the theory of biogenesis. Living cells arise only from pre-existing living cells. It changed medicine forever. If microbes do not just appear, but are transmitted, then we can stop them. We can wash our hands, we can sterilize our instruments, we can pasteurize our milk. The invisible world has rules, and because of Pasteur, we began to learn how to follow them. As you drift deeper into relaxation, consider the magnitude of this shift. For thousands of years, humanity lived in ignorance of the causes of disease and spoilage. We were blind to the ecosystem that operates on our skin and in our gut. Today, we have the window of the microscope. We have the map of the cell. We have defined our subject. We have polished our lenses. We have established that life is continuous, passing from one cell to the next in an unbroken chain, stretching back billions of years. You are now standing at the entrance of this vast, microscopic landscape. In our next lesson, we will step through the glass. We will move from the history of the tools to the architecture of the life they reveal. We will examine the difference between the ancient, simple cells of bacteria and the complex, structured cells that make up your own body. Let this introduction settle in your memory. The concept of magnification, the clarity of resolution, the discovery of the animalcule, the swan neck flask. These are the pillars upon which our knowledge rests. Lesson 2. Prokaryotic versus Eukaryotic cell structure. In our first lesson, we built the window through which we observe the world to the microscope. We established that life is not spontaneous. It is a continuous chain of inheritance. Now, we must look at the links in that chain. We must examine the fundamental unit of life, the cell. As you lie there, resting, visualize the cells in your own body. They are working tirelessly, consuming energy, repairing damage, and sending signals. But the cells that make up a human being are vastly different from the cells of the bacteria we studied in the previous lesson. In the grand scheme of biology, there is a great divide. Every living thing on this planet, from the smallest germ to the largest blue whale, falls into one of two primary categories based on how their cells are built. These two categories are the prokaryotes and the eukaryotes. Let us begin with the etymology, for the names reveal the secret. The word comes from the Greek word karyon, which means nut or kernel. In biology, this kernel refers to the nucleus, the dense central structure that holds the genetic blueprint. The first category is the prokaryote. Pro means before. These are the organisms that existed before the nucleus evolved. They are the before kernel cells. This group includes all bacteria and the ancient archaea. The second category is the eukaryote. Eu means true or good. These are the organisms possessing a true kernel or a true nucleus. This group includes you. It includes all animals, plants, fungi, and protozoa. Let us deepen our understanding of the prokaryote first. Visualize a prokaryotic cell. It is incredibly small, usually between 1 and 10 micrometers in size. It is ancient, rugged, and efficient. Think of a prokaryote like a one-room studio apartment. In a studio apartment, there are no walls separating the kitchen from the bedroom or the living room. Everything happens in one open shared space. In the bacterial cell, this open space is the cytoplasm. Floating within this jelly-like substance is the most precious treasure of the organism, its DNA. But unlike in your cells, this DNA is not locked away in a vault. It floats freely in a region called the nucleoid. The DNA is usually a single, circular loop of genetic instructions, naked and accessible. Because the prokaryote is like a studio apartment, it lacks internal rooms. It has no membrane-bound organelles. It has no mitochondria to generate power. It has no Golgi apparatus to package proteins. It must perform all the functions of life, generating energy, making proteins, and replicating right there in the open cytoplasm or along its outer membrane. This simplicity allows bacteria to reproduce at incredible speeds. They are stripped-down survival machines. Now, let that image fade and replace it with the image of a eukaryote. If the prokaryote is a studio apartment, the eukaryotic cell is a sprawling mansion. It is generally much larger, 10 to 100 times the size of a bacterium. It is complex, compartmentalized, and highly organized. The defining feature of this mansion is the master suite, the nucleus. In a eukaryotic cell, the DNA is not a simple loop. It is organized into multiple, linear chromosomes. To protect this vast library of information, the cell wraps it in a double layer of membrane called the nuclear envelope. This is the true kernel. The DNA is safe inside, separated from the chaos of the rest of the cell. But the complexity does not stop at the nucleus. The eukaryotic cytoplasm is filled with specialized structures called organelles. Think of these as the separate rooms of the mansion, each with a specific job. Visualize the mitochondria. These are the power plants of the cell. They take sugar and oxygen and convert them into ATP, the energy currency of life. Visualize the endoplasmic reticulum, or ER. It creates a maze of tunnels near the nucleus. This is the manufacturing floor where proteins and lipids are constructed. Visualize the Golgi apparatus. It looks like a stack of flattened pancakes. This is the post office. It receives proteins from the ER, tags them, packages them, and ships them to their destination. Visualize the lysosomes. These are the recycling centers, filled with digestive enzymes that break down waste and old parts. This compartmentalization allows eukaryotic cells to be incredibly complex and specialized, eventually allowing them to come together to build multicellular organisms like humans. But how did this leap occur? How did life go from the simple studio apartment to the complex mansion? The answer lies in one of the most fascinating theories in all of science, the endosymbiotic theory. Let us travel back in time, billions of years. The world is ruled by simple, prokaryotic life. The theory suggests that a large, predatory prokaryote engulfed a smaller prokaryote. It tried to eat it. But for some reason, the digestion failed. The small bacterium survived inside the larger one. Instead of being digested, the small bacterium began to help its host. It had a special talent. It could use oxygen to generate massive amounts of energy. It became a power plant. The host provided protection and food, and the small guest provided energy. They formed a partnership or symbiosis. Over millions of years, they became inseparable. That small, trapped bacterium evolved into what we now know as the mitochondrion. This is not just a story. The evidence is inside you right now. If you look closely at a mitochondrion, you will find it has its own DNA, separate from the nucleus. And that DNA is circular, just like a bacterium's. It has its own ribosomes, which are small and dense, just like bacterial ribosomes. Your cells are essentially ancient cooperatives, a merger of different organisms that learn to live as one. Understanding this difference between prokaryotes and eukaryotes is not just academic trivia. It is the foundation of modern medicine. It is the reason why you can take an antibiotic and survive. When you have a bacterial infection, you swallow a pill. That pill dissolves and spreads through your body. How does the drug know to kill the bacteria but leave your own cells alone? It relies on selective toxicity. Because bacteria are prokaryotes, their machinery is different from ours. For example, bacteria have a rigid cell wall made of a substance called peptidoglycan. Your cells do not have a cell wall at all. You are soft and flexible. Penicillin works by destroying peptidoglycan. It smashes the walls of the bacterial studio apartment, causing the bacterium to burst. But since your mansion cells don't have those walls, the drug bounces off harmlessly. Another example lies in the ribosomes. Ribosomes are the tiny machines that build proteins. Bacterial ribosomes are smaller and lighter, called 70S, than eukaryotic ribosomes, called 80S. Some antibiotics work by jamming the gears of the 70S ribosomes, starving the bacteria of proteins. Your heavier 80S ribosomes keep humming along, unaffected. So, as you lie there, realize that your body is a fortress of eukaryotic complexity. You are built of trillions of true kernels, each protecting your genetic code in a secure vault. You are defending yourself against the before kernels, the ancient, rapid, wall-building prokaryotes that seek to colonize you. This is the fundamental divide of biology. On one side, the swift and simple bacteria. On the other, the complex and compartmentalized eukaryote. We have now mapped the interior of the cell. We understand the machinery, but to truly identify our microbial neighbors, we must look at the exterior. We must look at their shape. In our next lesson, we will examine the morphology of bacteria, the spheres, the rods, and the spirals, and the colored stains we use to tell them apart. Let the concept of the Great Divide settle in your mind. The studio apartment and the mansion, the nucleoid and the nucleus, the simple and the complex. Rest in the knowledge that your cells are working perfectly, powering your rest with their ancient, symbiotic engines. Lesson 3. Bacterial morphology and cell walls. In our previous sessions, we built our microscope and we peered inside the cell to understand the complex machinery of life. We drew a line in the sand between the ancient prokaryotes and the complex eukaryotes. But tonight, we must refine our view. Imagine for a moment that you are a doctor, or perhaps a detective. You are presented with a patient who is ill. You suspect a bacterial infection. You take a sample, place it on a glass slide, and look through the lens. You know it is a bacteria, a prokaryote, but that is not enough information. There are millions of species of bacteria. To fight the enemy, you must identify it. You must know its face. This brings us to the study of morphology. Morphology simply means the study of form and structure. Just as we classify animals by their shape, identifying a bird by its wings or a fish by its fins, we classify bacteria by their shape and the armor they wear. As you relax, visualize the vast, dark void of the microscopic slide. Into this void, we introduce the three fundamental shapes of the bacterial world. The first shape is the coccus. The word comes from the Greek for berry, and that is exactly what they look like. Cocci are perfect spheres, round, smooth, and symmetrical. Imagine a singular, microscopic ball floating in the fluid, but these berries rarely exist alone. They are social creatures, and the way they group together tells us their specific name. If the spheres cling together in pairs, we call them diplococci. If they form long, elegant chains, like a pearl necklace strung across the slide, we call them streptococci. You may recognize that name from strep throat. If they bunch together in irregular grape-like clusters, we call them staphylococci. This is the staph in a staph infection. So, when you hear staphylococcus, you can now visualize the reality: a berry-shaped bacterium living in a grape-like cluster. The second shape is the bacillus. Bacillus means little staff or walking stick. These are rod-shaped bacteria. They look like microscopic capsules, or perhaps like a grain of rice or a tic-tac. They are longer than they are wide. Like the cocci, they can form chains, marching end to end across your field of view. This is the shape of many common organisms, from the E. coli living in your gut to the Bacillus anthracis that causes anthrax. The third shape is the spirillum. These are the elegant movers of the bacterial world. They are curved or spiral-shaped, looking like a corkscrew or a helical spring. This shape is not just for aesthetics, it is functional. Many spiral bacteria move by twisting, boring their way through viscous fluids like a drill bit entering wood. This is the shape of the bacterium that causes syphilis, and the shape of Borrelia, the cause of Lyme disease. Let those three shapes settle in your mind's eye: the sphere, the rod, the spiral. If you can identify the shape, you have taken the first step in solving the mystery. But shape is not enough. The world is filled with spheres and rods. To truly identify a bacterium, and more importantly, to know how to kill it, we must look at its armor. We must examine the cell wall. Almost all bacteria are encased in a rigid cell wall. This wall prevents the cell from bursting due to internal water pressure. It is made of a unique substance found nowhere else in nature, a mesh-like structure called peptidoglycan. Think of peptidoglycan as a strong, woven basket made of sugars and proteins that surrounds the delicate cell membrane. However, not all baskets are woven the same way. This difference in the cell wall is the most critical distinction in all of bacteriology. It splits the bacterial world into two warring camps: the gram-positives and the gram-negatives. To see this difference, we cannot use our naked eyes. We must use a technique developed in 1884 by a Danish physician named Hans Christian Gram. It is called the Gram stain. Visualize the process. It is like dyeing fabric. Imagine we have a slide with clear, invisible bacteria on it. First, we flood the slide with a purple dye called crystal violet. All the bacteria, regardless of their wall type, soak up this dye and turn a deep, dark purple. Next, we add iodine. The iodine acts as a mordant affixer. It binds to the purple dye, forming large crystals trapped inside the cell walls. Everything is still purple. Now comes the critical step. We wash the slide with alcohol. This is the moment of truth. If the bacterium is gram-positive, it has a thick, towering layer of peptidoglycan. It is like a thick wool sweater. The alcohol dehydrates this thick wall, tightening the mesh and trapping the large purple crystals inside. The bacterium remains purple. But if the bacterium is gram-negative, its wall is different. It has only a very thin layer of peptidoglycan. However, it possesses something the positive cells do not: a second, outer membrane made of lipids, fats. When the alcohol hits the gram-negative cell, it dissolves that outer fatty membrane just like soap dissolves grease. Because the peptidoglycan layer underneath is so thin and the outer armor is melted away, the purple dye washes right out. The bacterium becomes clear again. It is invisible. To see it, we must add a final, contrasting dye, usually a pink or red dye called safranin. The clear, gram-negative cells soak up this pink dye. So, at the end of the process, we look through the microscope and see a colorful landscape. The gram-positive bacteria, like Staphylococcus or Streptococcus, appear as deep, rich purple spheres or rods. They are the ones with the thick, absorbent walls. The gram-negative bacteria, like E. coli or Salmonella, appear as delicate pink or red shapes. They are the ones with the thin walls and the protective outer membrane. Why does this color matter to a doctor? Why does it matter to you? It matters because that cell wall dictates how the bacteria interact with your body and with medicine. Gram-positive bacteria, with their exposed peptidoglycan walls, are often very sensitive to antibiotics like penicillin. Penicillin works by attacking the construction of that wall. It is like pulling the thread on a wool sweater. The whole structure unravels and the cell bursts. Gram-negative bacteria are different. They are the armored tanks of the microbial world. Remember that extra outer membrane we dissolved with alcohol? In the human body, that membrane acts as a shield. It blocks many antibiotics, including penicillin, from entering the cell. This makes gram-negative infections notoriously difficult to treat. Furthermore, that outer membrane contains a molecule called lipopolysaccharide, or LPS. You do not need to memorize the chemical formula, but you should know its other name, endotoxin. When a gram-negative bacterium dies and breaks apart, it releases this toxin into your blood. This toxin triggers a massive, chaotic immune response. It can lead to fever, inflammation, and a dangerous condition called septic shock, where blood pressure plummets. This is why the gram stain is often the very first test performed in a hospital. Before a doctor prescribes a drug, they need to know, is the enemy purple or pink? Is it gram-positive with a thick wall, or gram-negative with a toxic shield? Let us synthesize this lesson. We have the shape, the sphere, coccus, the rod, bacillus, the spiral, spirillum. We have the arrangement, the chain, strepto, the cluster, staphylo, and we have the chemical nature of the wall, the purple gram-positive and the pink gram-negative. Imagine a report coming back from the lab, gram-positive cocci in clusters. Your mind should now translate that instantly. You see purple spheres. You see them bunched like grapes. You know they have thick walls. You suspect Staphylococcus. You know penicillin might work. Or perhaps the report says gram-negative bacilli. You visualize pink rods. You see the thin wall and the dangerous outer membrane. You know to be careful of toxins. You know you need strong, specific drugs to penetrate that shield. The microscopic world is no longer a blur. It is a world of specific shapes and specific colors, each telling a story of survival and defense. We have identified the players. We know what they look like. But a single bacterium is rarely a threat. The danger and the power of microbiology lies in numbers. How does one cell become a million? How does an infection explode from a minor nuisance to a life-threatening condition? In our next lesson, we will watch these shapes come alive. We will observe the relentless mathematics of microbial growth. We will watch 1 become 2, 2 become 4, and 4 become a billion. But for now, let the image of the purple spheres and the pink rods float in the darkness behind your eyelids. Structured, classified, understood. Lesson 4. Microbial Growth and Nutrition. We have spent our time thus far observing the individual. We have looked at the single cell, describing its walls, its shape, and its internal machinery. But in the world of microbiology, the individual is rarely the story. The power of the microbial world lies not in size, but in numbers. When we speak of growth in humans or animals, we usually mean an increase in size. A child grows into an adult, a seedling grows into a tree. But when we speak of microbial growth, we are not talking about a single bacterium getting fatter or taller. We are talking about an increase in population. We are talking about the relentless multiplication of life. As you rest here, let us focus on the mechanism of this expansion. It is a process of elegant simplicity called binary fission. Visualize a single bacterial cell floating in a nutrient-rich fluid. It has sensed that resources are abundant. It decides to divide. First, the cell elongates, stretching out slightly. Inside, its DNA, that single circular chromosome we discussed, copies itself. Now there are two loops of DNA, one moving to each end of the stretched cell. Then, a ring of protein forms in the center of the cell, cinching it tight like a belt being pulled around a balloon. This is the septum. The belt tightens and tightens until the cell walls fuse and finally snap apart. Where there was once one, there are now two. These two cells are identical clones. This process is not sexual, it is a mechanical duplication. And it happens fast. For a common bacterium like E. coli, this entire cycle can take as little as 20 minutes. Let that sink in. 20 minutes. Imagine you start with one cell. 20 minutes later, you have two. 20 minutes after that, 4, then 8, 16, 32. This is called exponential growth. It starts slowly, almost imperceptibly, but it accelerates with terrifying speed. If you allowed E. coli to grow unchecked for just 24 hours, the resulting mass of bacteria would weigh more than an aircraft carrier. But, thankfully, this infinite explosion does not happen in the real world. Bacteria do not live in a fantasy of unlimited resources. They live in a world of limits. When we grow bacteria in a closed container, like a test tube in a lab, or perhaps within an infection in the body, their population follows a predictable drama consisting of 4 distinct acts. We call this the growth curve. Let us walk through this story together. Act 1 is the lag phase. Imagine we have just introduced a few bacteria into a fresh tube of broth. You might expect them to start dividing immediately, but they do not. The population number remains flat. This is the lag phase. But do not be deceived. The cells are not sleeping. They are waking up. They are sensing their new environment. They are chemically tooling up, synthesizing the specific enzymes and proteins they need to digest the specific nutrients around them. It is a period of intense preparation, like an army gathering supplies before a march. Act 2 is the log phase, or the logarithmic growth phase. This is the explosion. The preparation is done. The cells begin to divide via binary fission at their maximum biological speed. 1 becomes 2, 2 becomes 4, millions become billions. The line on the graph shoots upward almost vertically. In this phase, the bacteria are most metabolically active. They are consuming nutrients voraciously. Interestingly, this is also the phase where they are most vulnerable to antibiotics. Drugs like penicillin, which attack the cell wall, work best here because the bacteria are busy building new walls. They are expanding so fast that their defenses are lowered. Act 3 is the stationary phase. The party cannot last forever. In the closed environment of the test tube, the nutrients, the sugar and proteins begin to run out. Space becomes crowded, oxygen levels drop. Furthermore, the waste products of billions of cells begin to accumulate, turning the environment acidic or toxic. The growth rate slows down. The number of new cells being born roughly equals the number of old cells dying. The curve on the graph flattens out into a plateau. The population has hit the carrying capacity of its environment. The bacteria initiate survival mode, halting their rapid division to conserve energy. Act 4 is the death phase. The resources are gone. The environment has become a soup of metabolic waste and toxins. The number of cells dying now far outstrips the number of new cells being formed. The curve dips downward. The population crashes. Some bacteria might dissolve completely. Others might form protective shells to wait out the apocalypse. It is the inevitable collapse of a population that has consumed everything. This cycle, lag, log, stationary, death, is the rhythm of microbial life. It happens in the soil, in the ocean, and in the food you eat. This brings us to the conditions that fuel this cycle. Bacteria are picky. They require specific environmental conditions to thrive, and the most critical of these is temperature. We classify microbes based on their Goldilocks zone, the temperature range where they grow best. First, visualize the cold, the psychrophiles. These are the cold lovers. They thrive in the icy waters of the Arctic or the depths of the ocean. Their proteins are flexible, designed not to freeze. They grow happily at temperatures that would freeze a human to death. You will rarely encounter these as pathogens, for your body is a tropical jungle to them. Next, visualize the heat. The thermophiles. These are the heat lovers. You find them in the steaming water of hot springs in Yellowstone, or in compost piles that generate their own heat. Their proteins are tough and rigid, designed not to unravel in the scorching temperatures. Between these two extremes lie the mesophiles. Meso means middle. These organisms love moderate temperatures, ranging roughly from 20 to 45 degrees Celsius. This range includes room temperature, and more importantly, it includes 37 degrees Celsius human body temperature. Most of the bacteria that cause disease in humans are mesophiles. They have evolved to view a warm human body as the perfect incubator. When you get a fever, your body is attempting to raise its internal temperature just enough to push these mesophiles out of their comfort zone, slowing down their enzymes and buying your immune system time to fight back. Understanding these temperature requirements provides the scientific basis for one of the most common appliances in your home, the refrigerator. Think about why we put milk in the fridge. We are not killing the bacteria. Cold is rarely a sterilization method. Instead, we are using temperature to manipulate the growth curve. By lowering the temperature to 4 degrees Celsius, we are taking the mesophilic bacteria that might be in the milk bacteria that would normally spoil it in hours and forcing them into a state of sluggishness. We are essentially freezing them in the lag phase, or a very slow log phase. Their molecular machinery slows down. They divide once every few days instead of every 20 minutes. We are not stopping time, but we are slowing the biological clock significantly. Conversely, this is why leaving food out on the counter is dangerous. We call the temperature range between 4 degrees and 60 degrees Celsius the danger zone. In this zone, mesophiles enter that rapid log phase. A single bacterium on a piece of chicken left at room temperature can become millions within a few hours, producing enough toxins to cause severe illness. Food safety is simply the practical application of the microbial growth curve. So, as you visualize these populations rising and falling, understand that life is a balance of resources and conditions. The bacterium is a machine designed to replicate, but it is bound by the laws of physics and chemistry. It needs fuel, it needs space, it needs the right warmth. We have observed how they grow. We have seen the mechanics of the population explosion, but growth requires work, and work requires energy. Where does this energy come from? How does a tiny cell, lacking a mouth or a stomach, consume the world around it? In our next lesson, we will descend to the molecular level. We will explore the engine of the cell. We will discuss metabolism, the chemical fires that burn within every living thing, turning sugar into the energy that powers the expansion of life. Let the phases settle in your mind: the lag, the log, the stationary, the death, the cold psychrophile, and the warm mesophile. See the curve rising and falling in the darkness. Rest now, knowing that your own body is maintaining its perfect temperature, keeping the balance, protecting you as you sleep. Lesson 5. Microbial Metabolism. We have watched the cell build its walls. We have watched the population expand in the curve of growth. But movement, reproduction, and repair all require one fundamental thing: energy. Just as a steam engine requires coal, or a city requires electricity, the microscopic city of the cell requires fuel. In this lesson, we descend into the boiler room of life. we will explore the complex, fiery chemistry that keeps the lights on. This is the study of metabolism. At its simplest level, metabolism is the sum of every single chemical reaction occurring within an organism. It is a constant cycle of destruction and creation. This cycle is divided into two opposing forces. The first is catabolism. This is the breakdown. It is the process of taking large, complex molecules like sugars or fats and smashing them apart to release the energy trapped inside their bonds. The second force is anabolism. This is the building up. It is the use of that released energy to construct the cell's own structures, building new walls, new proteins, and new DNA. The cell is constantly eating to build and breaking to create. But how does the energy move from the food to the machinery? It uses a molecular currency. Imagine a tiny, molecular, rechargeable battery floating in the cytoplasm. This molecule is called adenosine triphosphate, or ATP. The tri stands for 3. It holds 3 phosphate groups in a row. The bond holding the third phosphate is like a compressed spring. It is vibrating with potential energy. When the cell needs power, perhaps to move a flagellum or pump a nutrient, it snaps that third phosphate off. The spring releases its energy, and the ATP becomes ADP, adenosine diphosphate. The battery is dead. To recharge it, the cell must find fuel, break it down, and use that energy to reattach the phosphate, compressing the spring once again. The universal fuel for this process is glucose. Let us visualize the journey of a single glucose molecule as it enters a bacterium. This is a sugar molecule, a ring of 6 carbon atoms holding hands. The cell wants the energy hiding in those handshakes. The first step of the breakdown is called glycolysis. The word literally means sugar splitting. In the cytoplasm, a team of 10 enzymes attacks the glucose ring. They snap it in half, creating 2 smaller molecules called pyruvate. This snapping process releases a tiny burst of energy, enough to charge 2 ATP batteries. It is a start, but it is not much. The real treasure is still locked inside the pyruvate. Now, the cell faces a choice. The path it takes depends on one critical ingredient: oxygen. If oxygen is present, the cell can engage in the most efficient energy process known to biology: aerobic respiration. The pyruvate is ushered into a special cycle. In eukaryotic cells, this happens inside the mitochondria. In bacteria, it happens along the cell membrane. This cycle is often called the Krebs cycle. Here, the carbon molecules are stripped apart one by one. The carbon atoms are discarded as waste. This is the carbon dioxide you exhale with every breath. But the cell doesn't want the carbon, it wants the electrons. As the bonds break, high-energy electrons are stripped away and loaded onto carrier molecules. These carriers are like trucks, transporting the electrons to the final stage, the electron transport chain. Visualize a series of protein turbines embedded in the membrane. The carrier trucks arrive and dump their high-energy electrons into these turbines. As the electrons flow through the chain, they spin the turbines, which pump protons across the membrane, building up massive pressure. Finally, this pressure is released through a master turbine called ATP synthase, which spins wildly to recharge massive amounts of ATP. From a single molecule of glucose, aerobic respiration can generate up to 38 molecules of ATP. It is an incredibly efficient bonfire. But there is a catch. The electrons flowing through the chain must go somewhere at the end. If they pile up, the system clogs and stops. This is where oxygen enters the story. Oxygen sits at the end of the assembly line. It is the final electron acceptor. It catches the used electrons and combines with protons to form water, H2O. This is the only reason you breathe. You breathe to provide a trash collector for the electrons flowing through your mitochondria. Without oxygen, the chain stops, ATP production ceases, and the cell dies. But what if there is no oxygen? Many microbes live in places where air cannot reach, deep in the mud, inside your gut, or in a sealed container. They cannot use the efficient bonfire of respiration. They must rely on a backup plan. This plan is called fermentation. Fermentation is a survival strategy. Remember the first step, glycolysis? It produced a tiny amount of energy, 2 ATP, and split the sugar into pyruvate. Without oxygen, the cell cannot send that pyruvate to the Krebs cycle or the transport chain. Those doors are locked. So, the cell stops right there. It takes the pyruvate and converts it into a waste product, just to keep the glycolysis machinery running. This process is inefficient. Instead of 38 ATP, the cell only gets the initial 2. It is like burning a log only enough to char the bark, rather than burning it to white ash. Because the combustion is incomplete, plenty of energy remains left over in the waste products. These waste products are familiar to you. If a yeast cell ferments sugar, it transforms the pyruvate into ethanol, alcohol, and carbon dioxide. If a muscle cell or certain bacteria ferment sugar, they transform the pyruvate into lactic acid. Let that concept settle. Respiration is a complete burn, turning sugar into water and air. Fermentation is a partial burn, leaving behind chemical byproducts like alcohol and acid. While this seems simple, the microbial world is vastly more creative than humans. We have discussed organisms that eat sugar. These are called heterotrophs. But there is a group of microbes that defies our intuition. They are called lithotrophs. Litho means stone. These are the rock eaters. Deep underground, far from the light of the sun, miles beneath the surface where no organic food exists, these bacteria thrive. They do not eat sugar. They eat inorganic rocks and minerals. They strip electrons from sulfur, hydrogen, gas, or iron. They run a metabolism that is completely alien to our own, powering their lives by rusting the earth around them. They remind us that life is tenacious. It does not need sunlight or sugar. It only needs a flow of electrons. But let us return to the surface, to the world of humans. How does this invisible chemistry affect your life? The answer lies in the grocery store. The entire aisle of fermented foods is a display of microbial metabolism. Consider a loaf of bread. To make it, a baker mixes flour, sugar, and yeast. Saccharomyces cerevisiae. The yeast begins to eat the sugar. It engages in fermentation. As it breaks down the sugar, it releases carbon dioxide gas as a waste product. This gas gets trapped in the elastic dough, forming bubbles. These bubbles expand, causing the bread to rise. The small amount of alcohol produced burns off in the oven. You are eating the architectural result of fungal exhalations. Consider a tub of yogurt or a wheel of cheese. These begin as milk, which contains a sugar called lactose. We add bacteria like lactobacillus. These bacteria ferment the lactose, but they don't produce alcohol. They produce lactic acid. This acid sours the milk, causing the proteins to curdle and thicken into yogurt or cheese. The tang of cheddar is the taste of bacterial acid. Consider wine and beer. Here, we invite the yeast to feast on grape juice or grain water. We seal the vessel to cut off the oxygen, forcing them into fermentation. They consume the sugar and excrete ethanol. The alcohol we drink is simply the metabolic waste of a yeast cell trying to survive without air. Civilization was built on these metabolic tricks. Long before we knew what a bacterium was, we were harnessing their enzymes to preserve our food and brew our drinks. So, as you visualize the cell, see it as a furnace. It takes in fuel, sugar, fat, or even stone. It strips the electrons. It pumps the protons. It spins the turbines. It charges the batteries of ATP. This fire burns in every cell of your body right now. It burns in the soil beneath your home. It burns in the depths of the ocean. It is the universal hum of life, the conversion of matter into motion. Energy is the currency, but currency is useless without a plan on how to spend it. A battery can power a light bulb or a saw, The energy is the same, but the instruction is different. The cell needs a blueprint. It needs a manual. In our next lesson, we will open the library of the cell. We will look at the master code, the DNA. We will explore microbial genetics and learn how bacteria can not only read their own code, but steal code from others to gain new superpowers. Let the chemistry fade, the splitting of sugar, the flow of electrons, the rise of the bread. Lesson 6. Microbial Genetics. We have explored the body of the cell, its walls, its shape, and its movements. We have explored the engine of the cell, the fiery metabolism that burns sugar into energy. Now, we must explore the mind of the cell. How does a bacterium know how to build a cell wall? How does it know which enzymes to produce to digest sugar? How does it pass these instructions to its offspring? The answer lies in the most elegant molecule in the universe, deoxyribonucleic acid, or DNA. This is the study of genetics. It is the study of information. In the human world, if you want to build a cathedral, you need a set of blueprints. In the biological world, the DNA is that blueprint. But the way a bacterium stores this blueprint is quite different from how you store yours. Visualize the nucleus of a human cell. It contains 46 distinct chromosomes, linear strands of DNA, organized, wrapped, and paired. It is like a vast library of 46 separate volumes. Now, visualize the bacterial cell. Remember, it is a prokaryote. It has no nucleus. Its library is not a room of books, but a single, continuous scroll. Most bacteria possess a single, circular chromosome. This loop of DNA is twisted and coiled tight like a rubber band, floating in the cytoplasm in a region called the nucleoid. It contains all the essential information the bacterium needs to survive. It is the master list of essential files. But information is useless if it just sits there. It must be read. This process follows a universal rule known as the central dogma of biology. Let us trace the flow of command. The DNA is the master architect. It holds the plan, but it never leaves the safety of the center. When the cell needs to build a specific protein, say, an enzyme, to digest lactose, it does not send the whole DNA scroll to the construction site. Instead, it makes a copy of just that one specific page. This copying process is called transcription. The cell rewrites the DNA code into a temporary message made of RNA. This RNA acts as the foreman. It takes the instructions from the architect and carries them to the construction site. The construction site is the ribosome. Here, the final step occurs: translation. The ribosome reads the RNA message and builds the protein, brick by brick, amino acid by amino acid. DNA to RNA, RNA to protein. This is the flow of life, identical in you and in the germ. However, bacteria have a trick that humans do not. Alongside their massive main chromosome, they often carry tiny, extra loops of DNA. These are called plasmids. Think of the main chromosome as the operating system of a computer, essential, complex, and rarely changed. Think of the plasmids as apps or expansion packs. They are small, holding only a few genes. These genes are not necessary for day-to-day survival, but they offer special powers. One plasmid might carry the instruction for super strength, antibiotic resistance. Another might carry the instruction for toxic breath, toxin production. What makes bacteria truly terrifying and truly fascinating is their ability to share these apps. In the animal kingdom, genetic traits are passed vertically from parent to child. You have your mother's eyes because she passed those genes down to you. You cannot walk up to your friend, shake their hand, and suddenly acquire their eye color, but bacteria can. They engage in horizontal gene transfer. They can pass genetic instructions sideways, to their neighbors, to their friends, even to strangers of a different species. There are three primary ways they achieve this information trade. The first method is called transformation. This is the process of picking up naked DNA from the environment. To understand this, we must look back to 1928 and a British bacteriologist named Frederick Griffith. He was studying Streptococcus pneumoniae, a bacterium that causes pneumonia. He noticed there were two types, a rough strain that was harmless and a smooth strain that was deadly because it had a protective capsule. Griffith did something strange. He took the deadly smooth bacteria and boiled them, killing them completely. He injected these dead bacteria into a mouse. The mouse lived, of course. The bacteria were dead. Then he took the harmless rough bacteria and injected them. The mouse lived. Finally, he mixed the dead, deadly bacteria with the living, harmless bacteria and injected the mixture. The mouse died. When Griffith examined the mouse, he found living, deadly bacteria. How did this happen? The living, harmless bacteria had found the broken DNA fragments of the dead, deadly bacteria floating in the solution. They picked up the gene for the protective capsule, downloaded it, and integrated it into their own genome. They transformed themselves. They looted the bodies of the dead to acquire their weapons. The second method is called conjugation. This is often described as bacterial sex, though it is strictly for the transfer of information, not reproduction. Visualize two bacteria floating near each other. One contains a plasmid, a special superpower card. It wants to share it. This donor bacterium grows a long, hollow tube called a pilus. It looks like a grappling hook. It shoots this pilus out and latches onto a neighbor. It reels the neighbor in, pulling the two cells close together until their walls touch. Through this bridge, the donor sends a copy of the plasmid. It slides across the pilus like a message in a pneumatic tube. The connection breaks, and now both bacteria have the superpower. If that plasmid contained a gene for penicillin resistance, the recipient has instantly gone from vulnerable to invincible. The third method is called transduction. This is a case of accidental delivery. It involves a third party, a virus. There are viruses called bacteriophages that hunt bacteria. They land on a bacterium, inject their own DNA, and force the cell to make new viruses. Sometimes, in the chaos of assembly, a mistake happens. When the new viruses are being packed, one of them might accidentally pack up a piece of the bacterium's DNA instead of the viral DNA. It is like a letter being put in the wrong envelope. This virus bursts out and flies to a new bacterium. It injects its cargo, but the cargo isn't a viral attack. It's a gift of bacterial DNA from the previous victim. The new bacterium accepts this DNA and adds it to its library. These three mechanisms, transformation, looting the dead, conjugation, direct trade, and transduction, viral error, mean that the microbial world is constantly sharing secrets. This is the root of the crisis we face in modern medicine, antibiotic resistance. Imagine a hospital. A patient has an infection. Among the billions of bacteria in their body, perhaps only one has randomly mutated to be resistant to antibiotics. We treat the patient with antibiotics. The drug kills 99.99% of the bacteria. The competition is wiped out. But that one resistant survivor, it now has the entire playground to itself. It has all the food and all the space. It enters the log phase of growth. Soon, the patient is filled with resistant bacteria, but it gets worse. Through conjugation, these resistant survivors can extend their pili and pass the resistance plasmid to other species of bacteria in the gut or on the skin. The shield spreads like a rumor in a crowded room. This is why microbiology is not just about identifying the name of a germ. It is about understanding the flow of its intelligence. We are not fighting a static enemy. We are fighting a fluid, adapting, learning network. The bacterium is a master of information management. It keeps its essential files on the main chromosome safe and sound. It keeps its tactical files on plasmids, ready to be swapped and traded. It reads the environment, transcribes the code, and builds the tools it needs to survive. We have spoken of the cell. We have spoken of the DNA. But we briefly mentioned a predator, a virus that hunts bacteria. What is this entity? It has no cell wall. It has no metabolism. It cannot reproduce on its own. It exists on the very edge of the definition of life. In our next lesson, we will leave the world of the living cell and enter the twilight zone of biology. We will study general virology. We will examine the structure of these geometric parasites and ask the fundamental question: if they are not alive, how do they kill? Let the concepts of the code settle in your mind: the circular scroll, the trading of plasmids, the bridge of the pilus, the mouse that died so we could understand. Lesson 7. General Virology. For the past 6 lessons, we have inhabited the world of the cell. We have walked the halls of the eukaryotic mansion and the open floor plan of the prokaryotic studio. We have studied organisms that eat, breathe, grow, and reproduce. We have studied life. But now, we reach the edge of the map. We step into the grey area of biology, a twilight zone where the definitions of living and non-living begin to blur. We are here to study the virus. To understand a virus, you must first unlearn what you know about cells. A virus is not a cell. It has no wall. It has no cytoplasm. It has no mitochondria to burn fuel, no ribosomes to build proteins, and no nucleus to hold its secrets. It is stripped of all the machinery we associate with being alive. So, what is it? At its most fundamental level, a virus is simply information wrapped in a delivery package. It is a set of genetic instructions floating in the wind, waiting for an opportunity to be read. The scientific definition is precise and chilling. A virus is an obligate intracellular parasite. Let us dismantle that phrase. Parasite means it benefits at the expense of another. Intracellular means it must be inside a cell to function. Obligate means it has no choice. It cannot simply choose to live freely if it wants to. Without a host, it is nothing more than a complex speck of dust. As you rest, visualize the architecture of this speck. It is elegant in its simplicity. A virus consists of 2 absolute necessities. First, there is the genome. In your cells and in bacterial cells, the genetic picture is complete. You have DNA to store the code and RNA to read it. You have both. A virus, however, travels light. It possesses only one type of nucleic acid. It is either a DNA virus or an RNA virus, never both. This genome is the payload. It contains the instructions for making more viruses. That is its only purpose. It does not carry genes for digesting food or moving limbs. It carries only the blueprint for its own copy. Second, there is the capsid. This is the delivery box. The genome is delicate. It would easily be destroyed by the environment if it were left floating naked. So, the virus builds a protective shell around it, made of protein. This shell is constructed from identical protein subunits called capsomeres, which snap together like magnetic tiles to form a geometric shape. Nature favors symmetry, and viral capsids come in 3 primary shapes. Some are helical. Imagine a long, hollow tube, like a spring or a straw, with the genetic code coiled inside. This is the shape of the Ebola virus, a long, deadly thread. Some are polyhedral. These are many-sided geometric jewels. The most common is the icosahedron, a shape with 20 triangular faces and 12 corners. It looks like a microscopic soccer ball. The adenovirus, which causes sore throats, wears this shape. And some are complex. These are the most alien of all. The bacteriophage, the virus that eats bacteria, looks exactly like a lunar landing module. It has a geometric head, a central shaft, and spidery legs that act as landing gear. So we have the genome, the instructions, and the capsid, the box. For many viruses, this is enough. We call these naked viruses. They are rugged and tough. But there is a third component that some viruses possess, a component that adds a layer of deception. This is the envelope. Visualize the influenza virus or the coronavirus. These are not naked. When they leave the cell they have infected, they do not just leave, they steal a piece of the host's own cell membrane. They wrap themselves in this stolen coat. This outer layer is the envelope. It is made of lipids, fats, just like your own cells. Sticking out of this fatty envelope are protein spikes. If the envelope is the coat, these spikes are the keys. It is important to understand the scale of what we are discussing. We often lump germs together, thinking of bacteria and viruses as roughly the same size. This is incorrect. Imagine a bacterium is the size of a typical classroom. In this scale, a virus would be roughly the size of a soccer ball sitting on one of the desks. They are incredibly small. For centuries, scientists knew something was causing disease, but they couldn't see it. They would pass fluid through a filter, fine enough to catch every single bacterium, yet the fluid remained infectious. They called it a contagium vivum fluidum, a contagious living fluid. It wasn't until the invention of the electron microscope in the 1930 seconds that we finally saw the face of the enemy. But how does this tiny, static particle find its victim? It relies on specificity. A virus cannot infect just any cell. It is limited by its keys. The protein spikes on the surface of a virus are shaped to fit perfectly into specific receptors on the surface of a host cell. It is a lock and key mechanism. This is why your dog cannot give you the canine parvovirus. The keys on the parvovirus do not fit the locks on human cells. This is why you cannot get a tobacco mosaic virus from a plant. This is why hepatitis only attacks the liver and not the lungs. The virus drifts blindly until it bumps into a cell that matches its key. If the key fits, the door opens. If not, it simply bounces off and drifts away. This brings us to a practical realization about hygiene. We spoke of the envelope, that fatty layer stolen from the host. You might think that having an extra layer of protection makes a virus stronger, but ironically, it makes it weaker. Think about what you use to clean a greasy pan. You use soap. Soap dissolves fat. Because enveloped viruses like the flu, herpes, and coronavirus rely on that fatty outer coat to hold their keys, they are incredibly vulnerable to simple soap and alcohol. If you dissolve the fat, the envelope falls apart, the keys fall off, and the virus is neutralized. It is effectively dead. Naked viruses, however, like the norovirus, which causes stomach flu, have no fatty envelope. They are just a tough protein box. Alcohol sanitizers often fail against them because there is no fat to dissolve. This is why hand washing with the friction of water and soap is superior. It physically washes the rugged, naked viruses down the drain. Let us pause and consider the philosophical question that has plagued biologists for decades. Are viruses alive? They have DNA or RNA. They evolve. They mutate. They have a purpose. But if you put a virus in a sterile jar, it will sit there forever. It will not grow. It will not eat. It will not move. It is as inert as a grain of salt. It only becomes alive when it enters you. It borrows your energy. It borrows your ribosomes. It borrows your very existence to propagate its own. It is a hijacker. It turns the complex, beautiful machinery of the host cell into a factory for cheap viral copies. This is the nature of the viral existence. It is a borrowed life. As you lie there, safe in your bed, your body is constantly patrolling for these intruders. Your immune system is checking the keys of every particle it encounters. Do you belong here? Is your key a match for my lock? We have defined the particle. We see the genome, the dangerous letter. We see the capsid, the protective envelope. We see the spikes, the keys to the city. But a virus is only potential energy until the moment of contact. A letter is harmless until it is opened and read. In our next lesson, we will watch that letter open. We will observe the terrifying efficiency of viral replication. We will watch the virus attach, penetrate, and hijack the control center of the cell, turning the creation of life into an assembly line of destruction. Let the image of the geometric shapes float in the darkness. The icosahedron, the helix, the lunar lander, drifting, waiting, static. Lesson 8. Viral replication. In our last session, we examined the virus as an object. We held it in our minds as a static, geometric crystal, a box of protein holding a scroll of genetic code. We described it as potential energy, like a drawn bow or a loaded spring. A virus floating in the air or resting on a surface is inert. It does nothing. It simply waits. But the wait ends. The moment of contact arrives. In this lesson, we will animate the statue. We will watch the mechanism of infection unfold. This is the story of viral replication. It is a drama that plays out in 5 distinct acts, a cycle of invasion and conquest that turns the order of life into chaos. As you drift deeper into relaxation, visualize a single human cell. It is a bustling metropolis, unaware of the danger drifting toward it. The first act is attachment. The virus does not have eyes to see its target. It does not have fins to swim toward it. It moves by the random motion of physics, bumping into obstacles blindly. But eventually, it bumps into the right thing. Recall the protein spikes we discussed, the keys sticking out of the viral capsid. When the virus collides with the host cell, these keys probe the surface. They are looking for a specific receptor protein on the cell membrane. This is a moment of pure chemistry. If the shapes do not match, the virus falls away harmlessly. But if the key fits the lock, a chemical bond forms. The virus snaps into place. It is now tethered to the cell. It has docked. The second act is penetration and uncoating. The burglar is at the door. Now it must enter the house. The method of entry depends on the type of virus. Some viruses, particularly those with envelopes, use a technique called fusion. The fatty envelope of the virus melts into the fatty membrane of the cell, like 2 drops of oil merging into one. The capsid is dropped silently into the cytoplasm. Others, usually naked viruses, trick the cell into eating them. The cell perceives the virus as a nutrient and wraps its membrane around it, swallowing it whole in a bubble. Once inside, the virus must reveal its weapon. This is uncoating. The protein box is dissolved by enzymes, falling apart to reveal the naked viral genome. The blueprint is now loose in the factory. The third act is biosynthesis. This is the turning point. This is the hijacking. Up until this moment, the cell has been going about its business, making energy, repairing its walls, building its own proteins. But now, the viral genome takes command. It shouts orders to the cell's machinery. Stop making cell parts, it says. Make virus parts. The host cell's enzymes, confused but obedient, begin to read the viral code. They rush to copy the viral genetic material. They rush to the ribosomes to build viral proteins, thousands of capsids, thousands of spikes. During this phase, if you looked at the cell under a microscope, you might think the virus had disappeared. You wouldn't see any completed viruses. You would only see a frantic factory floor scattered with parts. This is often called the eclipse period. The virus has deconstructed itself to multiply. The cell is no longer serving its own survival. It is a zombie, working itself to death to build the army of its enemy. The fourth act is maturation. The parts are built. Now they must be put together. This is a marvel of self-assembly. The viral genomes find the empty protein capsids and slip inside. The capsomeres snap together, sealing the box. The spikes are arranged. It is like a factory assembly line running at high speed. One moment, you have a pile of protein and DNA. The next, you have thousands of perfectly formed infectious viral particles packed tight inside the dying host cell. The final act is release. The work is done. The cell is filled to bursting with new viruses. They need to get out to find new victims. There are two ways this ends. For naked viruses, the exit is violent. The cell becomes so packed with viruses that it loses its structural integrity. It produces an enzyme that eats its own wall from the inside. Suddenly, the cell bursts open. This is called lysis. The host cell dies instantly, and thousands of new viruses explode outward into the surroundings. For enveloped viruses, the exit is stealthier. They use a process called budding. The virus pushes against the inside of the cell membrane. It pushes and pushes until the membrane stretches around it and pinches off. The virus leaves, wrapped in a piece of the host's skin. The host cell does not pop, but it is slowly depleted, shedding viruses like a slow leak until it eventually withers and dies. This is the standard life cycle: attachment, penetration, biosynthesis, maturation, release. It is a cycle of immediate gratification and destruction. We call this the lytic cycle. But there is another way, a darker, quieter way. Some viruses, particularly certain bacteriophages and human retroviruses, are patient. They do not want to destroy the house immediately. They want to live in the attic. They choose the lysogenic cycle. In this scenario, the beginning is the same. The virus attaches and injects its DNA, but then something strange happens. Instead of taking over the factory, the viral DNA travels to the host's chromosome. It finds a spot and inserts itself right into the host's own genetic code. The virus has become a ghost. It is now called a prophage in bacteria or a provirus in humans. The host cell does not die. In fact, it might not even know it is infected. It goes back to its normal life. It eats, it grows, and most importantly, it divides. And here is the genius of the strategy. Every time the host cell divides, it copies its own DNA, and it unknowingly copies the viral DNA along with it. One infected cell becomes two, two become four, four become 1,000,000. The virus is spreading silently without ever making a single viral particle, simply by riding along in the host's lineage. This state of dormancy can last for generations. It can last for years. But eventually, something wakes it up. This awakening is called induction. Perhaps the host cell is stressed. Perhaps it is exposed to UV light, or chemicals, or starvation. The sleeping virus senses that the ship is sinking. It decides it is time to leave. The viral DNA snaps itself out of the host chromosome. It wakes up the machinery. It initiates the biosynthesis phase. The cell is suddenly thrown into chaos, filled with viruses and lysed. The sleeper agent has activated. This concept of lysogeny of the sleeping virus is not just a curiosity of bacteria. It is a reality of the human condition. We call it latency. Consider the virus varicella zoster. When you are a child, this virus causes chickenpox. You get the fever, the itchy spots, and then you recover. The virus seems to be gone. But it is not gone. The virus has traveled up the nerve fibers from your skin to the roots of your spinal nerves. There, it enters the nerve cells and becomes a provirus. It enters the lysogenic state. It goes to sleep. It can sleep there for 50 years. Your immune system cannot see it because it is hiding inside your own DNA. Then, one day, usually when you are older and your immune system is weaker or you are under great stress, the virus wakes up. Induction occurs. It travels back down the nerve fiber to the skin. It erupts in a painful, blistering rash. We call this shingles. It is the same virus re-emerging from its decades-long slumber. Consider the herpes simplex virus, which causes cold sores. It sleeps in the nerves of your face. Stress or sunlight wakes it up. causing a breakout before it retreats back into the shadows. This is the true power of the virus. It is not just a brute force attacker, it is a biological squatter. It can weave itself into the very fabric of your identity, becoming a permanent passenger in your cells. So, as you visualize the viral life cycle, do not just see the explosion, see the silence, see the viral code stitched into the host code, waiting for the signal. It is a reminder that in the microscopic world, the most successful predators are often the ones you cannot see. We have now explored the prokaryote, the eukaryote, and the virus. We have seen the diversity of the invisible world, but these organisms do not live in a vacuum. They are not content to stay on a glass slide. They are constantly seeking new territory, and often that territory is you. In our next lesson, we will return to the visible world. We will introduce the larger, more complex eukaryotic invaders. We will discuss the fungi, the yeasts and molds that decompose the world, and the protozoa, the ancient hunters that swim in the water and the blood. Lesson 9. The eukaryotic microbes, fungi and parasites. We have traveled a great distance in our microscopic journey. We began with the simple, ancient prokaryotes, the bacteria. We then examined the viruses, the non-living genetic hijackers. But now, we return to familiar territory. We return to the domain of the eukaryote. As you rest, recall the lesson on cell structure. You learned that you are a eukaryote. Your cells have a nucleus. They have mitochondria. They are complex mansions of biology. In this lesson, we will meet your microscopic cousins. These organisms share your cellular architecture, yet they live vastly different lives. They are the fungi and the protozoa. These are the organisms that bridge the gap between the microscopic and the macroscopic, ranging from single cells to visible networks that span entire forests. Let us begin with the fungi. When you hear the word fungus, your mind likely drifts to the mushroom growing on a damp log in the forest. But in microbiology, we are concerned with the fungi you cannot see, or at least the ones that start small. The kingdom fungi is a diverse group of organisms that are neither plants nor animals. Unlike plants, they cannot harvest energy from the sun, they have no chlorophyll. Unlike animals, they do not have mouths to ingest food. Instead, they are absorptive heterotrophs. They release powerful digestive enzymes out into their environment, onto the bread, onto the skin, onto the rotting leaf, digest the food externally, and then absorb the nutrients through their cell walls. Fungi are divided into two microscopic forms, yeasts and molds. Visualize the yeast. A yeast is a unicellular fungus. If you looked at it under the microscope, you would see a large, oval shape, far bigger than a bacterium. It is smooth and quiet. Yeasts do not usually divide by binary fission like bacteria. Instead, they reproduce by budding. Imagine a small bubble blowing out from the side of the parent cell. It swells, fills with cytoplasm and a copy of the nucleus, and then pinches off. This is the daughter cell. The most famous yeast is Saccharomyces cerevisiae, the sugar fungus of beer. It is the benevolent worker that gives us bread and wine. But there are others, like Candida albicans, that dwell in the human body. Usually, Candida is a harmless resident, kept in check by your immune system and your bacterial flora. But if that balance is disrupted perhaps by antibiotics, the yeast can overgrow, causing thrush or systemic infections. Now, visualize the mold. While yeast is a single cell, mold is a multicellular colony. It creates a fuzzy, cotton-like growth. This fuzz is actually a complex network of microscopic tubes called hyphae. Think of hyphae like the roots of a tree, but they don't just go down, they go everywhere. They weave together to form a visible mat called a mycelium. This is what you see on a piece of old bread. Molds reproduce by creating spores. Unlike the durable endospores of bacteria which are for survival, fungal spores are for reproduction. A single mold colony can release millions of lightweight spores into the air. They float on the wind like dust. If you leave a window open, you are inviting these spores in. When a spore lands on a moist, nutrient-rich surface, like a damp wall or a piece of fruit, it germinates, sending out its first tiny hyphae to colonize the new world. The cell wall of a fungus is a marvel of engineering. It is reinforced with chitin, the same tough, flexible substance that makes up the exoskeletons of insects and crabs. This makes fungal infections difficult to treat. Because their cells are so similar to yours, both are eukaryotic drugs that kill fungi often damage human cells as well. We have to target the few differences, like the specific sterols in their membranes, to treat a fungal infection without poisoning the patient. Now, let us shift our focus to the second group of eukaryotic microbes, the protozoa. The word protozoa means first animal. These are the hunters of the microscopic world. They are unicellular eukaryotes, but unlike fungi, they lack a rigid cell wall. This makes them soft, flexible, and capable of incredible movement. They live in water and soil, and a few of them live in us. We classify these first animals by how they move. Visualize the water of a pond teeming with life. First, we have the amoebas. The amoeba is a shapeshifter. It moves by extending a part of its own body forward. These extensions are called pseudopods, or false feet. The amoeba pushes its cytoplasm into the false foot, and the rest of the body flows behind it. It is a slow, oozing, rolling motion. The amoeba uses these false feet not just for travel, but for feeding. It reaches out, wraps its body around a food particle or a human cell, and engulfs it. This is a primitive, terrifying embrace called phagocytosis. While many amoebas are harmless, Entamoeba histolytica is a pathogen that can dissolve the lining of the human intestine, causing dysentery. Second, we have the flagellates. These organisms are built for speed. They possess long, whip-like tails called flagella. By lashing these tails, they propel themselves through fluids like a torpedo. Some are blood parasites, like trypanosoma, which causes African sleeping sickness. It swims through the bloodstream, evading the immune system by constantly changing its coat. Another is Giardia, a peculiar-looking parasite with two nuclei that look like eyes, giving it the appearance of a tiny, staring ghost. It lives in clear mountain streams, waiting for a hiker to drink the water, where it will then attach to the intestinal wall. Third, we have the ciliates. These are the hairy ones. Their entire body is covered in thousands of tiny, beating hairs called cilia. The cilia beat in perfect rhythm, like the oars of a Viking ship, allowing the ciliate to glide smoothly and turn with precision. The most famous example is the paramecium, the gentle giant of the pond water, grazing on bacteria. Very few ciliates cause disease in humans. They prefer the quiet life of the puddle. Finally, we have the apicomplexans. These are the most dangerous of all. They are non-motile. They cannot swim or crawl on their own. They are obligate parasites. They rely on a vector, an insect, to carry them from host to host. The king of this group is Plasmodium, the parasite that causes malaria. The life cycle of Plasmodium is a complex tragedy involving two hosts. It begins in the gut of a female Anopheles mosquito. When she bites a human, she injects the parasite into the bloodstream. The parasites travel to the liver, multiply, and then burst out to invade red blood cells. Inside the red blood cell, the parasite eats the hemoglobin, destroying the cell from the inside. Every 48 to 72 hours, millions of infected blood cells burst simultaneously, releasing waves of parasites and toxins. This cycle causes the rhythmic, bone-shaking fevers characteristic of malaria. To survive in the harsh world outside a host, many protozoa have developed a dual life strategy. When times are good, when food is plentiful and moisture is high, they exist as trophozoites. This is the feeding stage. They are active, swimming, and eating. But if the pond dries up, or if they are excreted from the body, they curl into a ball and secrete a tough, protective shell. This is the cyst stage. The cyst is dormant. It acts like a bacterial endospore. It can survive in dry soil, in chlorinated water, or on surfaces for months. When you drink contaminated water, you are swallowing the cyst. Once it reaches the safety of your intestines, the shell dissolves and the active trophozoite emerges to feed. Why does this matter to you? It matters because these organisms are the great recyclers and the great equalizers. The fungi are the saprobes. Without their digestive enzymes, the world would be buried under miles of dead trees and leaves. They break down complex organic matter, returning carbon and nitrogen to the soil so that new life can grow. They are the stomach of the earth. The protozoa are the base of the food web. They keep bacterial populations in check and provide food for small animals, which in turn feed the fish that feed us. But they also remind us of our vulnerability. Fungal infections, or mycoses, are notoriously difficult to cure. A bacterial infection might clear up in a week with antibiotics. A fungal infection of the nail or lung can take months or years to eradicate. The parasite Toxoplasma gondii, carried by house cats, quietly infects 1/3 of the human population, resting as cysts in our brains, mostly harmless, but always there. We are not alone in our bodies. We are ecosystems. We are defended by bacteria. We are challenged by viruses. We are colonized by fungi. We are hunted by protozoa. This concludes our survey of the microbial actors. We have met the entire cast: the prokaryote, the virus, the eukaryote. Now, the question arises: how do we control them? How do we keep the bread from molding, the wound from festering, and the water from killing us? In our next lesson, we will turn to the science of defense. We will explore control of microbial growth. We will learn the difference between sterilization and disinfection, and we will discover how heat, pressure, and chemistry can hold back the tide of the invisible world. Let the images of these complex creatures settle: the budding yeast, the spreading mold, the flowing amoeba, the complex cycle of the malaria parasite. They are complex, beautiful, and dangerous, but you are safe here. Rest in the knowledge of the world within worlds. Lesson 10: Control of microbial growth. We have spent 9 lessons exploring the vitality of the microscopic world. We have seen how bacteria grow with explosive speed. We have seen how viruses hijack the machinery of life. We have seen how fungi and parasites colonize their environments. The picture we have painted is one of relentless, expanding life. But human civilization relies on our ability to hold back this tide. Medicine, agriculture, and food preservation all depend on a single, critical concept: control. In this lesson, we will learn how to stop the invisible. We will study the physical and chemical methods used to reduce, inhibit, or destroy microbial populations. This is the science of making things safe. To begin, we must be precise with our language. In everyday conversation, we use words like clean or germ-free loosely. In microbiology, these words have strict definitions. The hierarchy of cleanliness is absolute. At the very top of this hierarchy is sterilization. Let that word carry its full weight. Sterilization is an absolute term. There is no such thing as slightly sterile or mostly sterile. Something is either sterile or it is not. Sterilization means the complete destruction or removal of all forms of microbial life. This includes vegetative bacteria, viruses, fungi, and most importantly, bacterial endospores. Recall that endospores are the survival bunkers of bacteria, tough, dormant shells that can withstand boiling water and harsh chemicals. Only sterilization destroys them. If a surgeon's scalpel is sterile, it is devoid of life. One step down from sterilization is disinfection. Disinfection is the destruction of vegetative pathogens, the active, living bacteria, but it does not necessarily kill endospores. It makes an object safe to handle, but it is not technically sterile. Crucially, the term disinfection applies only to inanimate objects. You disinfect a table, you disinfect a floor, you disinfect a surgical instrument, but you cannot disinfect a person. Living tissue is too fragile for the harsh chemicals used on floors. For living tissue, we use the term antisepsis. When a nurse wipes your arm with alcohol before an injection, this is antisepsis. The chemical is called an antiseptic. It reduces the microbial load on the skin to a safe level, but it does not sterilize your skin. To sterilize your skin would be to burn it off. So, how do we achieve these states? How do we kill the unkillable? The most common and effective weapon in our arsenal is heat. Heat works by denaturing proteins. Visualize the complex, folded proteins that make up the enzymes of a bacterium. They are like tangled balls of yarn, held together by delicate bonds. Heat vibrates these molecules until the bonds snap, the yarn unravels, the shape is lost. And since function follows shape, the enzyme stops working, the bacterium dies. But not all heat is created equal. There is moist heat, and there is dry heat. Moist heat, like boiling water or steam, is far more effective than dry heat. Water carries heat energy into the cell much faster than air. Think of the difference between reaching your hand into a 100-degree oven versus reaching your hand into a pot of 100-degree boiling water. The air feels warm, the water burns instantly. However, simple boiling is not enough to achieve true sterilization. Boiling water reaches 100 degrees Celsius. This kills most pathogens, but some resilient viruses and those tough bacterial endospores can survive boiling for hours. To kill them, we need to go hotter, but water boils at 100 degrees. It physically cannot get hotter than that, unless we change the rules of physics. We change the rules by adding pressure. This brings us to the single most important device in microbiology, the autoclave. Visualize a heavy, steel chamber with a thick door that locks tight. This is the autoclave. It is essentially a giant industrial pressure cooker. By sealing the chamber and pumping in steam, we raise the pressure. As the pressure rises, the boiling point of water rises. In a standard autoclave cycle, we raise the pressure to 15 pounds per square inch. This allows the steam to reach 121 degrees Celsius without boiling away. At this intense temperature, even the heat-resistant endospores are destroyed. The standard recipe for sterilization is simple: 121 degrees Celsius at 15 pounds per square inches for 15 minutes. If you place your glassware or your surgical tools in that chamber, they emerge completely sterile. But what if we don't want to sterilize? What if we just want to make food safe to drink? If we boiled milk, it would taste terrible. The proteins would curdle, the flavor would be ruined. To solve this, we turn to pasteurization, named after Louis Pasteur. This is a technique of gentle heat. We heat the liquid milk, juice, or wine just enough to kill the specific pathogens that cause spoilage and disease, but not enough to destroy the food itself. We are not sterilizing the milk. We are simply lowering the microbial count to safe levels. This is why milk eventually spoils in your fridge. The survivors eventually grow back. Heat is a powerful tool, but some things cannot be heated. You cannot autoclave a plastic syringe. It would melt. You cannot boil a delicate vaccine. It would be destroyed. For these heat-sensitive materials, we use filtration. Visualize a membrane with holes so tiny that water can pass through, but bacteria cannot. It is like a sieve. We pour the liquid through the filter, and the microbes are trapped on the surface. The liquid that drips out the other side is sterile. This is how we purify air in operating rooms and isolation wards, using HEPA filters to catch airborne pathogens. We also use the power of radiation. There are two types. First, there is ionizing radiation, like gamma rays and X-rays. These high-energy beams pass right through barriers. When they hit a cell, they shatter the DNA backbone. It is like taking a hammer to the library of the cell. We use this to sterilize plastic medical supplies, syringes, gloves, and sutures while they are still inside their packaging. Second, there is non-ionizing radiation, or UV light. Ultraviolet light is less penetrating. It damages DNA by causing adjacent letters to fuse together, creating thymine dimers that confuse the reading machinery. UV light is excellent for sterilizing surfaces and air, which is why you see that eerie blue glow in laboratory safety cabinets. We have spoken of physical methods: heat, pressure, filtration, and radiation. Now, let us turn to the chemical methods. Humanity has sought chemical shields for centuries. We use disinfectants and antiseptics to disrupt cell membranes or poison bacterial enzymes. Consider the phenols. These were the first surgical antiseptics used by Joseph Lister. They are powerful but irritating. Today, we use derivatives like the active ingredient in Lysol. They work by destroying the cell membrane, causing the bacterium to leak its contents. Consider the halogens. Iodine and chlorine are the heavy hitters here. Chlorine is the guardian of our water supply. A tiny amount of bleach, sodium hypochlorite, in the water system kills pathogens by oxidizing their molecules, essentially chemically burning them. Iodine is the gold standard for skin antisepsis. That brown-orange liquid painted on a patient's skin before surgery is betadine, an iodine compound that lingers and protects. Consider the alcohols. Rubbing alcohol, isopropanol, or ethanol is a staple of every first aid kit. Interestingly, pure 100% alcohol is not very effective. Alcohol needs water to work. A 70% solution is perfect. The water helps the alcohol penetrate the cell wall and denature the proteins inside. Alcohol evaporates quickly, leaving no residue, which makes it perfect for degerming skin before a needle stick. And finally, consider the surfactants, the soaps and detergents. Soap is not a killer. It has very little antiseptic value on its own. Its power is mechanical. Soap molecules have a unique structure. One end loves water, and the other end loves oil. When you wash your hands, the soap lifts the oil and dirt, and the bacteria trapped within them off your skin. The water then washes it all away. This process is called degerming. It is the physical removal of microbes rather than their destruction. And yet, it is arguably the single most effective public health measure in human history. So, as you visualize the battle against the invisible, understand that we have tools. We have the autoclave, the iron stomach that digests endospores with steam. We have the filter, the microscopic gatekeeper. We have the blue light of UV radiation. We have the chemical burn of chlorine and iodine. But notice something about all these methods. They are indiscriminate. Heat kills human cells just as fast as bacterial cells. Bleach is toxic to us. Radiation is dangerous. These controls are for the outside world. We use them on tables, on instruments, and on the surface of the skin. But what happens when the bacteria are already inside? How do we kill a pathogen floating in your bloodstream without killing you? We cannot boil a patient. We cannot pour bleach into their veins. We need a magic bullet. We need a substance that hunts only the bacteria and ignores the human. This was the dream of the early microbiologists. In our next lesson, we will see that dream realized. We will explore the era of antimicrobial drugs. We will learn how a forgotten petri dish led to the discovery of penicillin and how the war between humans and microbes shifted forever. Let the concept of sterility settle, the absolute clean, the pressure of the steam, the scrub of the soap. Lesson 11. Antimicrobial drugs. We have reached a pivotal moment in our curriculum. In our previous lesson, we discussed the crude tools of destruction: fire, steam, and harsh chemicals like bleach. These are effective weapons, but they are blunt instruments. They destroy everything they touch. If you were to drink bleach to cure a sore throat, you would kill the bacteria, but you would also kill yourself. For thousands of years, this was the limit of medicine. If an infection entered the bloodstream, there was little a doctor could do but offer comfort and wait. The human body had to fight alone. But the dream of a magic bullet always existed. This was a theoretical drug that could hunt through the body like a guided missile, finding and killing the pathogen while leaving the human host completely unharmed. This concept is known as selective toxicity. It is the holy grail of pharmacology. And in the 20th century, we found it. The story of this discovery is one of the great accidents of history. It begins in London in 1928 in the messy laboratory of a Scottish bacteriologist named Alexander Fleming. Visualize the scene. Fleming was studying Staphylococcus aureus, the bacteria that causes skin infections. He was known for being somewhat disorganized. Before leaving for a 2-week vacation, he stacked his petri dishes in the corner of his lab bench, forgetting to throw them away. He left a window open. While he was gone, a microscopic spore floated in through that open window. It was a spore of a common mold, Penicillium notatum. It drifted through the air and landed on one of the neglected petri dishes. It settled into the nutrient jelly and began to grow. When Fleming returned, he picked up the dish to clean it, but he stopped. He noticed something strange. The mold had grown into a large, fuzzy colony. But all around the mold, the Staphylococcus bacteria were gone. There was a clear ring, a zone of inhibition, where no bacteria could survive. The mold was secreting a chemical weapon to defend itself. Fleming isolated this chemical and named it penicillin. He had discovered the first true antibiotic. It was not a poison made by man, it was a weapon made by nature. But how does it work? How does this molecule distinguish between you and the germ? To understand this, we must return to the lesson on cell structure. Remember the cell wall. Bacteria, particularly gram-positive bacteria, are encased in a rigid shell made of peptidoglycan. This shell protects them from the high internal pressure of their own cytoplasm. Without it, they are like overinflated balloons waiting to pop. Human cells do not have cell walls. We have flexible membranes. Penicillin attacks this specific difference. It targets the enzyme that bacteria use to build the crosslinks in their cell wall. Think of the bacterium as a bricklayer building a wall around itself. Penicillin steals the mortar. The bacterium continues to grow, adding new bricks, but without the mortar, the wall is structurally unsound. Eventually, the internal pressure becomes too great. The wall collapses, and the bacterium bursts open and dies. Because human cells have no walls and no peptidoglycan, penicillin floats past them without causing any harm. This is the elegance of selective toxicity. But the cell wall is not the only target. Other antibiotics aim for the machinery of life, the ribosome. Recall that ribosomes are the protein factories of the cell. Both humans and bacteria need them to survive. However, the bacterial ribosome is slightly smaller and shaped differently than the human ribosome. In scientific terms, bacteria have 70S ribosomes, while humans have 80S ribosomes. Drugs like tetracycline and erythromycin are designed to fit perfectly into the gears of the bacterial 70S ribosome. They jam the machine. The bacterium can no longer make proteins. It cannot repair itself. It cannot grow. It effectively starves to death. Meanwhile, your larger 80S ribosomes keep humming along, too big for the drug to jam. There are other targets as well. Sulfur drugs block the production of folic acid, a vitamin bacteria need to make DNA. Humans get folic acid from our diet, so we don't have the machinery the drug attacks. Fluoroquinolones, like ciprofloxacin, attack the enzyme that uncoils bacterial DNA for replication. If the DNA cannot uncoil, the cell cannot divide. For a few decades, it seemed we had won the war. We had a pill for every bug. Infection, which had been the leading cause of death for all of human history, became a minor inconvenience. But we were arrogant. We underestimated the enemy. We forgot that bacteria have been evolving for 3 billion years. They are masters of adaptation. As soon as we introduced these chemical weapons, the bacteria began to evolve shields. This is the crisis of antibiotic resistance. It is a simple case of Darwinian evolution. Imagine a colony of 1 billion bacteria infecting a patient. We administer an antibiotic. The drug kills 999,999,999 of them. But perhaps one bacterium has a random mutation. Maybe its cell wall is slightly different. Maybe its membrane pump is slightly stronger. This one bacterium survives. With its competitors dead, this survivor has all the food and space it needs. It multiplies. Within a day, the patient is filled with billions of bacteria, all descended from that one resistant mutant. The antibiotic no longer works. Bacteria have developed ingenious ways to fight back. Some produce enzymes that physically destroy the drug. The most famous is beta-lactamase, an enzyme that chews up the penicillin molecule before it can touch the cell wall. Some change the locks. They alter the shape of their ribosomes so the drug can no longer fit inside to jam the gears. Some build pumps. These are called efflux pumps. If the antibiotic manages to enter the bacterial cell, the bacterium instantly pumps it back out, like a bailing bucket on a sinking ship. This arms race is escalating. We now face superbugs like MRSA, methicillin-resistant Staphylococcus aureus, and VRE, vancomycin-resistant Enterococcus, that are immune to almost everything in our arsenal. We are entering a post-antibiotic era, where a simple scrape or a routine surgery could once again become life-threatening. This reality teaches us a profound lesson. We cannot rely solely on chemistry to save us. Drugs are temporary. They are static molecules fighting a dynamic, living enemy. Eventually, the enemy learns. But do not despair. While our pharmaceutical inventions are failing, we have a defense system that is far older and far more sophisticated than any drug. It is a system that learns. It is a system that remembers. It is a system that evolves alongside the enemy. This is your immune system. Unlike a pill which attacks blindly, your immune system is an intelligent army. It has scouts, snipers, heavy artillery, and historians. It does not just kill the pathogen. It takes a snapshot of its face so that it can never harm you again. So, as we close the chapter on drugs, we open the chapter on the body itself. In our next lesson, we will explore the first line of defense. We will look at the walls of the fortress, the skin, and the mucous membranes. We will meet the foot soldiers, the white blood cells that patrol your veins, ready to eat anything that does not belong. Let the history of the magic bullet settle in your mind. The accidental mold, the bursting wall, the jammed ribosome, the resistant survivor. Lesson 12. Innate Immunity. We have spent our time thus far studying the invaders. We have analyzed their shapes, their weapons, and their strategies. We have looked at the chemical warfare of antibiotics that humans invented to fight them. But drugs are a recent invention. For 99% of human history, we survived without penicillin. We survived because we are not defenseless. We are fortresses. You are currently inhabiting a body that is a masterpiece of defensive engineering. It is constantly under siege by the billions of microbes that live on your skin, in the air you breathe, and on the food you eat. Yet, you are rarely sick. This is due to your immune system. We divide this system into two great branches. The first branch, which we study tonight, is called innate immunity. The word innate means born with. This is the defense system you possessed the moment you took your first breath. It does not need to learn, it does not need to memorize the face of the enemy. It is non-specific. It treats every intruder roughly the same way. Whether it is a virus, a bacterium, or a fungus, the innate system sees it simply as non-self. It is the brute force security guard that stops anyone who does not show an ID badge. The innate system operates in two lines of defense. The first line of defense is preventing entry. It is the wall of the castle. The second line of defense is what happens when the wall is breached. It is the battle in the courtyard. Let us visualize the first line, the physical and chemical barriers. Your primary shield is the skin. We often think of skin as soft and sensitive, but to a bacterium, it is an impenetrable wasteland. The outer layer of your skin, the epidermis, is made of cells that are packed tight and filled with a tough, waterproof protein called keratin. Crucially, the top layers of these cells are dead. Think of the brilliance of this design. A virus needs a living cell to hijack. A bacterium needs nutrients to grow. But the surface of your body is a desert of dead, dry protein. Even if a pathogen lands there, it eventually falls off as you shed your skin cells. You shed millions of these rafts every day, carrying the hitchhikers away with them. Furthermore, your skin is chemically hostile. Your sweat is salty, creating a hypertonic environment that dehydrates bacteria. Your oils are slightly acidic, keeping the pH low enough to inhibit growth. But you are not just skin. You have openings, eyes, nose, mouth. These are the weak points. Here, the body deploys mucous membranes. Mucus is a sticky, thick trap. It captures dust and microbes before they can travel deeper into the lungs. Lining your respiratory tract are microscopic hairs called cilia. These hairs beat in a synchronized wave, constantly pushing the dirty mucus upward, away from the lungs and toward the throat, where you subconsciously swallow it. This is the ciliary escalator. It keeps the lungs sterile. And what happens when you swallow that mucus? It drops into the pit of the stomach. The stomach is a vat of hydrochloric acid. With a pH of roughly 2, it is corrosive enough to dissolve metal. Very few microbes can survive a bath in stomach acid. This is why you can eat an apple without sterilizing it first. Your stomach incinerates the microbial load. Consider your eyes. They are constantly washed by tears. Tears contain a magical enzyme called lysozyme. This enzyme specifically attacks the chemical bonds in the peptidoglycan cell wall of bacteria. Every time you blink, you are washing your eyes with a natural antibiotic that causes bacteria to burst. However, walls can be broken. A cut, a scrape, or a puncture wound opens a gateway. The enemy enters the sterile tissue beneath. Now, the first line has failed. The body activates the second line of defense. This is a cellular counterattack. The alarm is sounded, and the soldiers of the blood are summoned. These soldiers are the white blood cells, or leukocytes. Specifically, we rely on a group called phagocytes. The word means eating cells. The first responder to the scene is the neutrophil. Neutrophils are the infantry. They are abundant, circulating in your blood by the millions. When a bacterium enters a wound, the damaged tissue releases chemical distress signals. The neutrophils smell this blood in the water, a process called chemotaxis. They squeeze through the walls of the blood vessels and rush to the site. Neutrophils are aggressive and suicidal. They gorge themselves on bacteria until they die. The pus you see in an infected wound is largely a graveyard of dead neutrophils that sacrifice themselves to hold the line. Behind them comes the heavy armor, the macrophage. The name literally means big eater. These are massive, shape-shifting cells that wander through your tissues like security guards on patrol. When a macrophage encounters a bacterium, it initiates the drama of phagocytosis. Visualize this process. The macrophage reaches out with long, fluid arms called pseudopods. It grabs the bacterium. The arms wrap around the invader, hugging it tight, and pull it inside the macrophage's body. The bacterium is now trapped inside a bubble, called a phagosome. It is still alive, trapped in a prison cell. But the macrophage has a weapon ready. Floating in its cytoplasm are organelles called lysosomes. These are bags filled with digestive enzymes and toxic oxygen radicals. The macrophage fuses the prison bubble with the acid bag. The phagosome becomes a phagolysosome. Inside this chamber of horrors, the bacterium is dissolved. It is digested into its raw chemical components. The macrophage effectively eats the enemy and spits out the debris. But this battle does not happen quietly. The battlefield itself changes. This is the phenomenon of inflammation. You know the signs of inflammation: redness, heat, swelling, and pain. These are not mistakes. They are tactical decisions made by your body. When tissue is damaged, special cells release a chemical called histamine. Histamine tells the nearby blood vessels to dilate to widen. This brings more blood to the area. This is why the wound turns red and feels hot. You want this heat. It speeds up the metabolism of your white blood cells and inhibits the growth of some pathogens. Histamine also makes the blood vessels leaky. The walls become permeable. This allows fluid to seep out of the blood and into the tissue. This causes swelling or edema. Why would you want swelling? Because that fluid contains clotting factors to seal the wound and antibodies to fight the infection. It helps wall off the infection to prevent it from spreading. And finally, the pain. The swelling presses on nerve endings, and chemicals stimulate pain receptors. This is a warning. It tells you, do not use this part. Let it rest. Let it heal. There is one final weapon in the innate arsenal, a weapon that affects the entire body: fever. When neutrophils and macrophages fight a large infection, they release chemical signals that travel to the brain, specifically to the hypothalamus, the body's thermostat. These signals tell the hypothalamus to reset the temperature. 37 degrees is too comfortable, the brain decides. Raise the heat to 39. You begin to shiver. This is your muscles generating heat. You feel cold because your internal set point is higher than your current temperature. Fever serves a purpose. It cooks the bacteria. Most pathogens are mesophiles. They love 37 degrees. At 39 or 40 degrees, their growth slows down. Their enzymes become sluggish. Simultaneously, the fever causes the liver to hoard iron, starving the bacteria of a vital nutrient. It is a scorched earth policy. The body makes itself an inhospitable wasteland to drive the invader out. So, realize that when you feel sick, when you are inflamed, feverish, and swollen, you are not feeling the disease. You are feeling the cure. You are feeling the energy of your own innate system waging a total war to keep you alive. This system is powerful. It is fast. It is brutal. But it has a flaw. It has no memory. A neutrophil kills a staph bacteria today. If you get infected by the same staph bacteria next year, the neutrophil won't remember it. It starts from scratch every single time. It cannot get better at its job. For true, lasting protection, we need a system that learns. We need a system that remembers the face of the enemy and builds a custom weapon to destroy it. We need the elite special forces. In our next lesson, we will meet the adaptive immune system. We will learn about the B cells and T cells, the antibodies, and the miracle of immunological memory that allows vaccines to work. Let the sensation of heat and protection settle in your mind. The wall of the skin, the trap of the mucus, the hunger of the macrophage, the fire of the fever. Lesson 13. Adaptive immunity. In our last lesson, we stood on the ramparts with the innate immune system. We watched the brute force of the fever and the indiscriminate hunger of the macrophage. That system is essential. It keeps you alive from moment to moment. But it is a blunt instrument. It treats every enemy the same. It does not learn and it does not remember. But there is another level of defense, a level so sophisticated that it can distinguish between two viruses that differ by a single atom, a level that can remember an infection you had 50 years ago and protect you from it today. This is the adaptive immune system. This system does not rush. It takes time to wake up, sometimes days or weeks. But when it does not just fight the enemy. It obliterates it with a precision that defies imagination. The adaptive system is built upon two pillars: specificity and memory. It relies on the ability to recognize antigens. An antigen is any molecule that triggers an immune response. It is the name tag of the pathogen. It might be a protein spike on a virus or a sugar molecule on the wall of a bacterium. While the innate system looks for general signs of trouble, the adaptive system reads these specific name tags. It doesn't just see bacteria, it sees Staphylococcus aureus, strain B. The soldiers of this system are the lymphocytes, the B cells, and the T cells. They are born in the bone marrow, but they lead very different lives. Let us first follow the journey of the humoral response. This is the domain of the B cell. The B stands for bone marrow, where these cells mature. Imagine a B cell floating in your bloodstream. On its surface, it is covered in thousands of tiny receptors. These receptors are Y-shaped proteins. But here is the miracle. Every single B cell in your body has a slightly different receptor shape. You have billions of B cells, representing a library of billions of possible shapes. They are waiting for the one specific key that fits their lock. One day, a specific bacteria enters your body. It floats past millions of B cells, and nothing happens. The shapes don't match. But then, it bumps into the one B cell that happens to have the perfect matching receptor. Click. The connection is made. This B cell wakes up. It has found its destiny. It begins to divide rapidly, cloning itself thousands of times. This army of clones then transforms into plasma cells. Plasma cells are factories. They do not fight directly. Instead, they pump out millions of copies of their specific receptor and release them into the blood. These free-floating receptors are called antibodies. An antibody is a Y-shaped protein weapon. It travels through the blood and lymph, seeking out the specific antigen it was made for. When it finds the bacteria, it attaches. This attachment is devastating for the invader. First, antibodies cause agglutination. Because the antibody has two arms, it can grab two different bacteria at once, gluing them together. This clumps the bacteria into a helpless, sticky ball that is easily eaten by macrophages. Second, they perform neutralization. If antibodies cover the spikes of a virus, the virus can no longer attach to your cells. It is wrapped in a straitjacket, rendered harmless. Third, they act as a beacon. A bacterium covered in antibodies lights up to the rest of the immune system like a Christmas tree, screaming, "Eat me!" This is the humoral response. It fights the enemy outside the cells, in the fluids or humors of the body. But what if the enemy is already inside? What if a virus has penetrated your cells and is hiding in the cytoplasm? The antibodies cannot reach it there. They cannot cross the cell membrane. For this, we need the assassins. We need the cell-mediated response. This is the domain of the T cell. The T stands for thymus, a small gland in your chest where these cells go to be trained. There are two main types of T cells, and they work together like a command unit. First, there is the helper T cell, CD4. This is the general of the immune army. It does not kill. It coordinates. When a macrophage eats a bacteria, it takes a piece of that bacteria, an antigen, and holds it up on its surface like a trophy. It shows this trophy to the helper T cell. The helper T cell sees the enemy intel and sounds the alarm. It releases chemical signals called cytokines. These signals wake up the B cells to make antibodies, and they wake up the macrophages to eat faster. Without the helper T cell, the entire immune system sleeps. This is why HIV is so deadly. The HIV virus specifically hunts and kills helper T cells, leaving the body without a commander. Second, there is the cytotoxic T cell, CD8. This is the sniper. This cell patrols the body, touching your own cells, checking their ID badges. It is looking for cells that have been infected. When a human cell is infected with a virus, it does something brave. It takes a piece of the virus and displays it on its own surface. It puts up a flag that says, "I am infected, kill me." The cytotoxic T cell sees this flag. It docks with the infected cell. It does not try to heal it. It is too late for that. Instead, it secretes a protein called perforin. Perforin punches a hole in the membrane of the infected cell. Then, the T cell shoots enzymes through that hole that trigger apoptosis-programmed cell death. The infected cell commits suicide, shriveling up and dying to prevent the virus from spreading. The T cell then detaches and moves on to find the next victim. This dance of B cells and T cells is powerful, but it takes time. The first time you encounter a pathogen, the primary response, it can take a week or two for the right cells to find the antigen and build an army. During that week, you get sick. The bacteria multiply. You fight the fever. But the true genius of the adaptive system lies in what happens next. After the battle is won, most of the clone soldiers die off. The swelling goes down, the energy returns, but a few cells remain. These are called memory cells. There are memory B cells and memory T cells. They do not die. They retreat to the lymph nodes and wait. They are veteran soldiers who know exactly what the enemy looks like. They can wait for decades. 10 years later, you inhale the same virus. The memory cells see it. They do not need a week to wake up. They remember. They activate instantly. Within hours, they produce a flood of antibodies so massive that the virus is destroyed before you even feel a tickle in your throat. You don't even know you were infected. You are immune. This concept, immunological memory, is the foundation of vaccination. When we vaccinate you, we are not giving you a disease. We are giving you a simulation. We inject a harmless piece of the pathogen, a dead virus, a single protein spike, or a piece of mRNA. Your innate system sees it and gets angry. Your adaptive system sees it and learns. It builds the antibodies. It trains the memory cells. Then the simulation ends. The antigen is cleared, but the memory cells remain. You have gained the experience of the war without the danger of the battle. When the real disease comes, the real polio, the real measles, your body is already an expert. It crushes the invasion instantly. This is the triumph of the adaptive immune system. It transforms your biological history into biological armor. Every cold you have ever fought, every vaccine you have ever received, has added a new volume to your library of defense. You are a walking archive of every victory you have ever won. So, as you rest, visualize these elite forces patrolling your blood, the Y-shaped antibodies, locking onto targets, the helper T cells, coordinating the flow of information, the cytotoxic T cells, quietly removing the compromised, and the memory cells sleeping in the archives, watching and waiting. Lesson 14. Epidemiology and Public Health. We have traveled a long road together. We began with the lens of the microscope, peering at the single, vibrating atom of a cell. We moved to the chaos of the infection, the heat of the fever, and the precision of the antibody. We have studied the war between the microbe and the man as it happens inside the body. But biology does not exist in a vacuum. You are not a solitary island. You are part of a community. You are part of a species. And just as microbes move from cell to cell, they also move from person to person, from city to city, and from continent to continent. This brings us to the grand conclusion of our curriculum: epidemiology. Epidemiology is the study of when and where diseases occur, and how they are transmitted in populations. It is the detective story of medicine. If the doctor treats the patient, the epidemiologist treats the world. They do not ask, what made this person sick? They ask, why did everyone in this neighborhood get sick on Tuesday? To understand this science, we must learn the language of the patterns. We classify diseases not by what they do to the body, but by how they behave in the crowd. Visualize a map of the world. First, consider the term endemic. An endemic disease is a constant presence. It is always there, lingering in the background at a steady, predictable level. The common cold is endemic. Malaria is endemic in parts of Africa. It is part of the landscape, a low-level static that never truly goes away. Next, consider the epidemic. This is a spike. It is a sudden, unexpected rise in the number of cases in a specific area. It is an outbreak. Imagine a calm ocean, and then suddenly a rogue wave rises up. That wave is the epidemic. It might be a flu outbreak in a school or a surge of Ebola in a village. It is an event that demands immediate attention. Finally, consider the pandemic. This is when the wave becomes a tsunami. A pandemic is an epidemic that has crossed international borders. It spans continents. It affects the globe. It is a reminder that in the biological world, there are no borders. A virus that evolves in a cave on one side of the world can reach a city on the other side within hours, carried on the wings of a jet plane. But how does the pathogen travel? It needs a path. We call this the chain of transmission. The chain begins with the reservoir. The reservoir is the home of the pathogen. It is where the germ lives, grows, and multiplies before it attacks. Sometimes the reservoir is human. We carry the measles. We carry the flu. Sometimes the reservoir is animal. We call these zoonoses. Rabies lives in the nervous system of a bat or a dog. The plague lives in the flea of a rat. And sometimes the reservoir is non-living. The soil itself can be a reservoir. The bacteria that cause tetanus and botulism live in the dirt, waiting for a puncture wound or a canning jar to wake them up. Water can be a reservoir for cholera. From the reservoir, the pathogen must move. This is transmission. It might be direct contact, a handshake, a kiss, or a sneeze. It might be indirect contact, touching a doorknob or a toy that a sick person touched hours ago. We call these inanimate objects fomites, or it might be carried by a vector. A vector is a living courier. Usually, it is an arthropod, the mosquito carrying malaria, the tick carrying Lyme disease, the flea carrying the plague. The vector is the bridge that allows the pathogen to leap from the wild reservoir into the human home. To truly understand the power of epidemiology, we must look back to the moment it was born. The year is 1854. The place is London. At the time, London was a crowded, filthy, industrial city. Cholera was ravaging the population. It is a terrifying disease, causing rapid dehydration and death within hours. No one knew what caused it. The leading theory of the time was miasma, the idea that disease was caused by bad air or foul smells rising from the rotting garbage in the streets. Enter John Snow. He was a physician, but he thought like a detective. When a massive outbreak of cholera struck the Soho district, killing hundreds of people in days, Snow did not look at the air. He looked at the habits of the victims. He took a map of London. For every death, he placed a black dot on the location of the victim's house. As he worked, a pattern emerged from the chaos. The black dots were not randomly scattered. They were clustered tightly around a single intersection, Broad Street, and right in the center of that cluster was a public water pump, the Broad Street pump. Snow interviewed the survivors. He found that almost everyone who died had drunk water from that specific pump. He found a woman who died miles away in a healthy neighborhood, only to discover she had liked the taste of the Broad Street water so much she had her son bottle it and bring it to her. Snow presented his map to the city officials. He didn't have a microscope. He didn't know what a bacterium was, but he knew the pattern. He convinced them to take a simple physical action: remove the handle from the pump. They did. The handle was removed, the water flow stopped, and the epidemic ended. This was the birth of modern public health. It proved that we do not need to see the germ to stop it. We just need to break the chain of transmission. This logic governs our world today. When you wash your hands, you are breaking the chain. When we treat our sewage, we are destroying the reservoir. When you wear a mask, you are blocking the portal of exit. But perhaps the most powerful concept in epidemiology is the idea of herd immunity. This is the shield of the community. Imagine a population where no one is immune to a disease. If one person gets sick, they infect 3 others. Those 3 infect 9. The fire spreads unchecked. Now, imagine a population where 90% of the people are immune, either because they survived the disease or, more likely, because they were vaccinated. The virus enters. It infects one person. That person sheds the virus, but everyone around them is immune. The virus hits a wall. It cannot find a new host. The chain of transmission is broken. The fire dies out because it has no fuel. Crucially, this protects the vulnerable. The newborn baby who is too young to be vaccinated, or the cancer patient whose immune system is weak, they are safe. They are protected not by their own bodies, but by the bodies of their neighbors. They are protected by the herd. This is why vaccination is considered a civic duty. It is not just about your health. It is about becoming a brick in the wall that protects the entire species. We have now reached the end of the curriculum. We have traversed the landscape of life. We started with the cell, the fundamental unit, the studio apartment, and the mansion. We examined the bacteria, the ancient masters of the planet, with their shapes and their walls. We watched them grow, doubling and doubling in the log phase. We watched them eat, burning sugar in the fires of metabolism. We read their genetics, the scrolling code of the chromosome and the trading of plasmids. We met the virus, the geometric hijacker that exists on the edge of life. We met the fungi and parasites, the recyclers and the hunters. We learned how to control them with the heat of the autoclave and the chemistry of the pill. We marveled at our own defense, the walls of the skin and the memory of the white blood cells. And finally, we stood back to see the patterns, the maps of disease, and the power of community. You now possess the foundational knowledge of microbiology. The invisible world is no longer a mystery to you. It is a system. It has rules, it has logic, and most importantly, it has balance. Microbes are not just our enemies. They are our ancestors. They are our digesters. They are the engineers of our atmosphere. We live in their world, and they live in us. We are a partnership. Lesson 15. Disorders of the immune system. We have spent the last few sessions marveling at the efficiency of the human immune system. We have described it as a fortress, a well-trained army, and a library of memory. We have seen how it identifies the foreign invader, neutralizes the threat, and remembers the encounter for a lifetime. When this system works correctly, it is the silent guardian of your health. You walk through a world teeming with pathogens, yet you remain well. But the immune system is a weapon, and like any weapon, it carries a risk. If it is aimed in the wrong direction, or if the trigger is pulled too easily, the protector becomes the destroyer. In this lesson, we will explore what happens when the immune system malfunctions. We will study the chaos of hypersensitivity, where the body attacks harmless substances, and the tragedy of autoimmunity, where the body attacks itself. Let us begin with the phenomenon of hypersensitivity. You likely know this by its common name, allergy. An allergy is fundamentally a mistake. It is a case of mistaken identity. Your immune system encounters a substance that is objectively harmless. A grain of pollen, a peanut protein, a speck of dust, or the dander of a cat. These substances are not viruses. They are not trying to hijack your cells. They are not bacteria trying to eat your tissue. They are inert. However, in an allergic individual, the immune system perceives these harmless particles as a mortal threat. It treats a grain of pollen as if it were a deadly parasite. To understand the mechanism of this mistake, we must look at a specific type of antibody called immunoglobulin E, or IgE. In a non-allergic person, IgE is rare. It is reserved for fighting actual parasites, like tapeworms. But in an allergic person, the body produces massive amounts of IgE in response to the allergen. Visualize the process of sensitization. Imagine you inhale ragweed pollen for the first time. Your body sees it and decides, incorrectly, that it is dangerous. Your B cells wake up and churn out millions of IgE antibodies specific to that pollen. But you don't feel sick yet. This is just the preparation. These IgE antibodies travel through your blood and attach themselves to the surface of special white blood cells called mast cells. Mast cells are abundant in your skin, your nose, and your lungs. They are like floating landmines. They are packed full of granules containing powerful chemicals. So, after that first exposure, you have millions of mast cells patrolling your body, primed and loaded with IgE, waiting for the pollen to return. Now, visualize the second exposure. You walk outside on a spring day. You inhale the pollen again. The microscopic grains drift into your nose and land on the mast cells. The pollen binds to the IgE antibodies sticking out of the cell surface. This is the trigger. Instantly, the mast cell undergoes degranulation. It explodes, releasing its payload of chemicals into the surrounding tissue. The most famous of these chemicals is histamine. Histamine is a powerful signaling molecule. It tells the body to flush out the invader. It causes vasodilation, the widening of blood vessels. This leads to redness and heat. It causes the vessels to become leaky, allowing fluid to rush into the tissue. This causes swelling and a runny nose. It stimulates nerve endings, causing the sensation of itching. It causes the smooth muscles in the lungs to contract, narrowing the airways to prevent the invader from going deeper. This is the wheezing of asthma. All of these symptoms, the sneezing, the itching, the congestion, are not caused by the pollen. They are caused by your own body's desperate, misguided attempt to wash the pollen away. Usually, this reaction is local, a runny nose, or a patch of hives, but sometimes the reaction is systemic. This is called anaphylaxis. If the allergen enters the bloodstream directly, perhaps through a bee sting or a peanut being digested, the mast cells explode all over the body simultaneously. The release of histamine is massive. All the blood vessels in the body dilate at once. This causes a catastrophic drop in blood pressure, known as shock. The brain and heart are starved of oxygen. Simultaneously, the airways constrict so tightly that breathing becomes impossible. This is a medical emergency. It requires the immediate injection of epinephrine, adrenaline, which forces the blood vessels to constrict and opens the airways, countering the effects of the histamine. Let that concept settle. The allergy is a false alarm that rings so loudly it can destroy the building. Now, we must turn to a darker malfunction. If allergy is the immune system attacking a harmless foreign object, autoimmunity is the immune system attacking the self. This is a failure of self-tolerance. During the training of your T cells in the thymus, there is a rigorous selection process. Any T cell that reacts to your own body proteins is destroyed. It is deleted. The body ensures that its soldiers know the uniform of the home team. But sometimes, a rogue cell escapes this screening. Or sometimes, a molecular mimicry occurs, where a virus looks so much like a human cell that the immune system gets confused and attacks both. The result is a civil war. The immune system turns its arsenal against the very tissues it was born to protect. Consider rheumatoid arthritis. Here, the immune system targets the synovium, the delicate lining of the joints. Antibodies attach to the joint tissue, and macrophages move in to eat the enemy. But the enemy is your own cartilage. The result is chronic, painful inflammation that eventually destroys the bone and fuses the joint. Consider multiple sclerosis, MS. Here, the target is the nervous system. Specifically, the T cells attack the myelin sheath. Myelin is the fatty insulation that wraps around your nerve fibers. It allows electrical signals to travel quickly from the brain to the muscles. In MS, the immune system strips this insulation away, leaving the nerves exposed and scarred. The electrical signals slow down or stop completely. The result is a progressive loss of sensation, movement, and coordination. The body is literally losing its connection to itself. Consider type 1 diabetes. This is not caused by eating too much sugar. It is an autoimmune assault on the pancreas. The immune system identifies the beta cells, the cells responsible for making insulin, as foreign invaders. The cytotoxic T cells hunt them down and destroy them. Without beta cells, the body cannot produce insulin, and it loses the ability to control blood sugar. Why is this happening? Epidemiologists have noticed a disturbing trend. Allergies and autoimmune diseases are becoming significantly more common in developed, wealthy nations. This observation led to the hygiene hypothesis. The theory suggests that our world has become too clean. For millions of years, the human immune system evolved to fight constant battles against worms, parasites, and bacteria. It is an aggressive army designed for a high-threat environment. But in the modern world, we have clean water, we have antibiotics, we have sanitized homes, we have eliminated the parasites. The army is still there, but it has no enemy to fight. Bored and understimulated, the immune system begins to look for trouble. It starts attacking pollen, it starts attacking dust, it starts attacking the thyroid or the pancreas. By eliminating the external enemies, we may have inadvertently turned the weapon inward. This teaches us a profound lesson about balance. Health is not simply the absence of disease. Health is a state of equilibrium. The immune system must be strong enough to kill the plague, but gentle enough to ignore the dust. It must be vicious enough to destroy a cancer cell, but wise enough to spare the nerve cell sitting next to it. It is a tightrope walk over a canyon. As you rest, consider the complexity of this regulation. Even now, as you sleep, your body is making millions of decisions. Is this me? Is this foreign? Ek. Do I attack? Do I ignore? Your survival depends on these microscopic choices being made correctly, millions of times a minute. We have seen the power of the system, and we have seen its potential for error. We have looked at the enemies, the bacteria, the viruses, the fungi. We have looked at the defenses, the fever, the antibody, the macrophage, and now we have looked at the disorders, the allergy, and the autoimmunity. This completes our clinical understanding of the host. But the story of microbiology is not just about the individual patient. It is about the environment. It is about the soil beneath your feet and the air in your lungs. There are microbes that do not care about humans at all. They are busy running the machinery of the planet. In our next lesson, we will step outside the hospital and into the wild. We will explore environmental microbiology. We will descend into the soil to watch bacteria turn death into life, recycling the elements that make up your body. Lesson 16. Microbial diseases of the skin and eyes. We have explored the internal workings of the immune system, the hidden wars fought in the blood and the bone marrow. Now, we move to the surface. We move to the boundary where the human body meets the outside world. This is the integumentary system, the skin. Your skin is an organ of impressive scale. It covers nearly 2 square meters of surface area. It is a waterproof, flexible, and self-repairing coat of armor. But in the eyes of a microbe, your skin is not just a wall, it is a vast and varied geographical landscape. To a bacterium, the dry forearm is a desert, the armpit is a humid tropical jungle, the oil glands are rich, deep caves filled with nutrients, and like any landscape on Earth, the skin is populated. You are covered in a normal flora, billions of bacteria that live on you peacefully, occupying space and preventing dangerous pathogens from finding a foothold. However, this peace is fragile. If the barrier is broken by a cut, a burn, or an insect bite, the neighbors can turn hostile. Or a transient invader can land and aggressively colonize the territory. In this lesson, we will examine the most common and the most dangerous infections of the skin and eyes. We begin with the two titans of skin infections, Staphylococcus and Streptococcus. Visualize Staphylococcus. You recall from our morphology lesson that these are gram-positive spheres that grow in grape-like clusters. The most important species here is Staphylococcus aureus. The name aureus means golden. When you grow this bacterium in the lab, the colonies often have a golden yellow pigment. This organism is the ultimate opportunist. It lives in the nasal passages of roughly 20% of the population, causing no harm. But if it enters a hair follicle, it triggers an intense local inflammation. This is a folliculitis, or a pimple. If the infection goes deeper, it becomes a furuncle, commonly known as a boil. This is a painful, pus-filled abscess where the body has walled off the bacteria with fibrin. If the body fails to contain it, the boils can fuse together into a massive, deep lesion called a carbuncle. At this stage, the patient often has a fever as the infection threatens to enter the bloodstream. What makes Street aureus so dangerous is its arsenal of toxins. It produces coagulase, an enzyme that clots the blood around the bacteria, building a fortress to hide from your immune system. Worse, it produces exfoliative toxin. In newborns, this toxin can cause scalded skin syndrome, where the outer layer of the skin peels off in sheets, as if the child had been burned. The second titan is Streptococcus, specifically Streptococcus pyogenes. These bacteria grow in long, elegant chains. Unlike staph, which often stays localized in a boil, strep loves to spread. It secretes enzymes that dissolve the glue holding your cells together, allowing the bacteria to sweep through the tissue like a wildfire. A common manifestation is impetigo, a highly contagious infection in children that causes crusty, honey-colored sores on the face. But if Streptococcus pyogenes reaches deeper tissue, it becomes the flesh-eating bacteria you hear about in horror stories. The medical term is necrotizing fasciitis. In this condition, the bacteria attack the fascia, the connective tissue sheath surrounding your muscles. The toxins destroy the tissue faster than the surgeon can cut it away. The immune system overreacts, causing massive inflammation that cuts off blood supply, leading to gangrene. It is a race against time, where amputation is often the only way to stop the spread. Now, let us shift our focus from the bacterial invaders to the viral ones. Many viral diseases are classified as exanthems, skin rashes caused by a virus that entered through the respiratory tract. The infection begins in the lungs, enters the blood, and then blooms on the skin. Consider the measles, rubeola. It begins with a fever and a runny nose, but the hallmark sign appears inside the mouth: Koplik spots, tiny white dots with red halos. Soon after, a macular rash erupts on the face and spreads down the body. While often dismissed as a childhood illness, measles is incredibly dangerous, capable of causing pneumonia and brain damage. It is one of the most contagious viruses known to science. If one person has it, 90% of susceptible people in the room will catch it. Consider rubella, or German measles. This is a milder rash, often overlooked, but its danger lies not to the patient, but to the unborn. If a pregnant woman catches rubella, the virus can cross the placenta and cause severe birth defects, known as congenital rubella syndrome. This is why the MMR vaccine, measles, mumps, rubella, is a cornerstone of public health. Consider chickenpox, varicella. We discussed this in the context of latency. The virus, varicella zoster, causes itchy, fluid-filled vesicles to pop up all over the skin. These vesicles eventually scab over. But remember, the disappearance of the rash is not the end of the war. The virus retreats to the spinal nerves, waiting for decades to re-emerge as shingles. Finally, we must look to the window of the soul, The eye. The eye is protected by the conjunctiva, a thin membrane lining the eyelid and covering the white of the eye. When this membrane becomes infected, we call it conjunctivitis or pink eye. It can be caused by bacteria like Haemophilus influenzae or by viruses like adenoviruses. The eye becomes red, swollen, and gritty. While uncomfortable, it is usually self-limiting. However, there is an eye infection that changes history. It is called trachoma, caused by the bacterium Chlamydia trachomatis. This is not a sexually transmitted disease in this context. It is spread by flies or dirty hands touching the eyes. Trachoma is the leading cause of infectious blindness in the world. The infection causes chronic inflammation of the eyelid. Over years of repeated infections, the inner eyelid becomes scarred. The scarring causes the eyelid to turn inward, a condition called trichiasis. When the eyelid turns in, the eyelashes turn in with it. The lashes scratch the sensitive cornea every time the patient blinks. Blink by blink, day by day, the cornea is abraded and scarred until it becomes opaque. The patient slowly goes blind, seemingly scratched into darkness by their own lashes. It is a tragedy because it is preventable with simple hygiene and antibiotics. This brings us to the real-world synthesis of this lesson. The diseases of the skin and eyes are largely diseases of contact. The Staphylococcus lives on the gym equipment. The Streptococcus lives on the unwashed hand. The trachoma lives on the towel shared between siblings. The most powerful medical intervention for these diseases is not a complex drug, it is soap. It is the physical removal of the pathogen before it can breach the wall. It is the covering of the wound to deny the staph entry. It is the washing of the face to keep the flies away from the eyes. Think of the vulnerability of the surface. Your skin is tough, but it is constantly besieged. Every time you scratch an itch, you are creating a micro breach. Every time you shave, you are opening the gates. But your body is ready. The salt is there. The acid is there. The white blood cells are waiting just beneath the surface. As you lie there, visualize your own skin. Feel the air against it. Understand that it is a sealed barrier, a seamless suit of armor that separates the clean interior from the microbial exterior. The Staphylococcus may be sitting on your skin right now, but it cannot get in. You are closed. You are safe. We have covered the outside, but the microbes have other ways to enter. They can bypass the skin entirely. They can enter through the food you eat or the air you breathe, or they can strike directly at the control center. In our next lesson, we will travel inward to the most protected and sensitive system of all. We will study microbial diseases of the nervous system. We will look at what happens when bacteria cross the blood-brain barrier to attack the mind itself. Lesson 17. Microbial diseases of the nervous system. We have journeyed from the outer walls of the skin to the inner sanctum of the human body. We have studied the defenses of the blood and the tissue, but there is one system that stands apart. It is the master controller, the seat of your consciousness, and the regulator of every breath you take. This is the nervous system. Because the brain and spinal cord are so critical, the body protects them with a ferocity seen nowhere else. The nervous system is encased in bone, the skull, and the vertebrae. But bone is not enough. Inside the bone, the brain is wrapped in 3 layers of tough, protective membranes called the meninges. Visualize these layers. The outer layer is the dura mater, the tough mother. It is thick and leather-like. Beneath that is the arachnoid mater, a spiderweb-like structure filled with fluid. And closest to the brain is the pia mater, the gentle mother, a delicate film that hugs every fold of the cerebral cortex. Between these layers flows the cerebrospinal fluid, CSF, a clear, sterile liquid that acts as a shock absorber. But the ultimate defense is chemical, not physical. It is the blood-brain barrier. In the rest of your body, the capillaries, the tiny blood vessels, are leaky. They have small gaps that allow water, nutrients, and white blood cells to drift easily into the tissues. But in the brain, the capillaries are sealed tight. The cells are stitched together with tight junctions. Nothing gets through unless the brain specifically opens a door for it. This barrier keeps out toxins, drugs, and most importantly, microbes. The brain is an immunologically privileged site. It cannot afford inflammation. Swelling in your hand is painful. Swelling inside the rigid box of your skull is fatal. Therefore, the immune system is largely barred from entering. This isolation is generally perfect, but when it fails, the results are catastrophic. We begin with the infection of the membranes themselves. Meningitis. Meningitis is not a specific germ. It is a condition. It is the inflammation of the meninges. It can be caused by viruses, bacteria, or fungi. Viral meningitis is common and usually mild. The patient gets a headache and a stiff neck, but they typically recover. Bacterial meningitis, however, is a medical emergency of the highest order. Imagine a bacterium like Neisseria meningitidis or Streptococcus pneumoniae. These organisms possess a thick capsule that allows them to slip past the body's initial defenses in the throat. They enter the bloodstream. Through mechanisms we still struggle to fully understand, they manage to trick or break through the blood-brain barrier. Once inside the cerebrospinal fluid, they find a paradise. The fluid is rich in nutrients, and because the white blood cells are locked out, there is no police force to stop them. The bacteria multiply explosively. The body reacts with panic. It forces the blood-brain barrier open to let immune cells in. This causes massive inflammation. But the skull is a closed container. It cannot expand. As the meninges swell, the pressure inside the skull rises. It presses on the brain tissue. The symptoms are classic and terrifying: a pounding headache, a fever, and the hallmark sign, a stiff neck. The pain is so severe that the patient cannot touch their chin to their chest. In the case of Neisseria meningitidis, the bacteria release a potent endotoxin that shatters blood vessels throughout the body. This causes a distinctive purple rash that does not fade when you press a glass against it. It is the sign of blood leaking under the skin. Without rapid antibiotic treatment, death can occur within hours. Now, let us move from the brain to the nerves. We must discuss two diseases caused by the same family of bacteria, yet they are polar opposites. They are the diseases of paralysis, tetanus and botulism. Both are caused by the genus Clostridium. These bacteria are obligate anaerobes. They cannot survive in oxygen. They live in the soil, deep in the mud, or inside the intestines of animals, surviving as dormant endospores. First, tetanus. The agent is Clostridium tetani. The reservoir is the soil. Imagine you step on a rusty nail while gardening. The nail pierces your foot, depositing dirt and spores deep into the tissue. The wound closes up. Inside, the environment is dark and oxygen-free. The spore wakes up. The bacteria do not spread through your body. They stay in the foot. But they release a neurotoxin called tetanospasmin. This toxin enters your nerve endings and begins to travel. It travels retrograde, backwards up the nerve fibers until it reaches the spinal cord. Normally, when you move a muscle, one set of muscles contracts while the opposing set relaxes. This relaxation is controlled by inhibitory neurotransmitters like glycine. Tetanospasmin destroys the release mechanism for these inhibitors. It cuts the brakes. The result is rigid paralysis. The muscles are told to contract, and they are never told to stop. The first sign is often in the jaw, lockjaw. The masseter muscles clench tight and cannot open. The paralysis spreads down the body. The back muscles spasm so violently that the spine can arch backward in a bow shape, a condition called opus totanos. The contraction is strong enough to break the patient's own vertebrae. Death follows when the diaphragm spasms, and the patient can no longer exhale. It is a horrific way to die, fully conscious but trapped in a prison of stone. Now, consider the opposite, botulism. The agent is Clostridium botulinum. This bacteria does not usually grow in the body. Instead, it grows in food. It loves anaerobic environments, like a sealed can of beans or a jar of preserved garlic. If the canning process is not hot enough to kill the spores, they survive. Inside the sealed jar, they germinate and secrete the botulinum toxin. This toxin is odorless and tasteless. When you eat the food, you ingest the poison. The toxin travels to the neuromuscular junction, the gap where the nerve meets the muscle. Normally, to trigger a muscle, the nerve releases a chemical called acetylcholine. Botulinum toxin acts as a wall. It blocks the release of acetylcholine. The signal is sent, but the message never arrives. The result is flaccid paralysis. The muscles go limp. It usually starts in the face. The eyelids droop. The speech becomes slurred, double tongue. The patient cannot swallow. The paralysis descends down the body. The arms hang uselessly. Finally, it reaches the diaphragm. The muscle needed to breathe simply stops working. The patient suffocates, not because they cannot exhale, but because they cannot inhale. Let that distinction settle. Tetanus is the failure to relax, a rigid, spastic death. Botulism is the failure to contract, a limp, silent death. Botulinum toxin is the most poisonous substance known to science. A single gram of crystalline toxin, evenly dispersed, could kill 1 million people, and yet humans have found a use for it. By injecting microscopic diluted amounts of this toxin into the face, we can paralyze the small muscles that cause wrinkles. We call this Botox. It is the same mechanism, flaccid paralysis of the forehead muscles. It is a testament to our strange relationship with microbes that we use the deadliest poison on earth for cosmetic vanity. There are other invaders of this system. We could speak of polio, the virus that hunts the motor neurons of the spinal cord, leaving children with withered limbs. We could speak of rabies, the slow-moving virus that climbs the nerves from a dog bite to the brain, turning the host into a hydrophobic, aggressive vector before killing them. All these diseases share a common theme. They attack the wiring. They disrupt the electrical flow that makes you you. This lesson serves as a stark reminder of our fragility. The nervous system is powerful, but it is delicate. It relies on precise chemistry, the release of glycine, the flow of acetylcholine, the pressure of the fluid. A single microscopic spore releasing a single molecular toxin can shatter that balance. However, we are not helpless. We have the tetanus vaccine, the T, in the DTaP shot, which teaches your body to neutralize the toxin before it touches a nerve. We have the polio vaccine, which has nearly driven the virus to extinction. We have canning regulations to prevent botulism. We have the blood-brain barrier, holding the line every second of every day. As you rest, visualize that barrier. See the tight junctions of the capillaries in your brain. They are sealed, they are vigilant, your thoughts are safe. Your dreams are protected. The control center is secure. We have now explored the skin and the nerves, but the body must breathe. It must eat. In our next lesson, we will enter the great open cavities of the body. We will explore the microbial diseases of the respiratory and digestive systems. We will look at the lungs, constantly filtering the air, and the gut, the most bacteria-filled place on Earth. Lesson 18. Microbial diseases of the respiratory and digestive systems. In our previous lesson, we visited the brain, a fortress locked away behind bone and barrier, isolated from the world. But the body cannot stay isolated. To survive, it must engage with the environment. You must bring the outside world in. Every minute of your life, you pull air deep into the delicate sponges of your lungs. Every day, you take physical matter from the earth, plants, animals, and water, and place it inside your body to be digested. The respiratory system and the digestive system are the great open gates of human biology. They are tubes that run through us, connecting the inner self to the outer atmosphere. And because they are open, they are the primary highways for infection. Let us begin with the breath, the respiratory system. The lungs are a paradise for microbes. They are warm, they are moist, and they are constantly supplied with fresh oxygen. The body defends them with mucus and cilia, but some invaders have evolved to bypass these traps. We must speak of the White Plague, tuberculosis. For centuries, this was the leading cause of death in the Western world. It was called consumption because it seemed to consume the patient from the inside out. The agent is a bacterium called Mycobacterium tuberculosis. It is a strange organism. It grows very slowly, sometimes taking 20 hours to divide just once. Its secret weapon is its wall. It does not have a standard cell wall. Instead, it is coated in a thick, waxy substance called mycolic acid. This wax makes it incredibly tough. It effectively wears a waterproof raincoat. Visualize the infection. You inhale a droplet containing the bacteria. It travels deep into the alveoli, the tiny air sacs of the lung. Your immune system sends a macrophage to eat it. The macrophage swallows the bacterium, but because of that waxy coat, it cannot digest it. The acid of the macrophage simply rolls off. The bacterium survives inside the immune cell, living there, multiplying slowly. The body realizes it cannot kill the invader, so it decides to imprison it. It sends more macrophages, more T cells, and more fibrous tissue to surround the infected area. It builds a wall of cells around the bacteria. This structure is called a tubercle. Inside this prison, the tissue dies and becomes cheese-like. Eventually, the body deposits calcium around it, calcifying the tomb. In a healthy person, the bacteria go to sleep inside this stone prison. This is latent TB. You are not sick, you are not contagious. But if your immune system weakens due to age, or HIV, or starvation, the prison walls crumble. The bacteria wake up. They break out of the tubercle and consume the lung tissue, leaving cavities and holes. The patient coughs up blood, and the cycle begins again. Now, let us look at the virus that changes its face every year: influenza. The flu is not a bad cold. It is a systemic assault caused by the influenza virus. To understand the flu, you must visualize the surface of the viral particle. It is covered in protein spikes. There are two types, and they are the reason for the confusing names you hear on the news, like H1N1 or H5N1. The H stands for hemagglutinin. These are the spikes the virus uses to attach to your cells. They are the keys. The N stands for neuraminidase. These are the spikes the virus uses to cut itself free from the cell after it has replicated. They are the scissors. The influenza virus is a master of disguise. It uses two strategies to evade your immune system. The first is antigenic drift. This is a slow accumulation of small mutations. The H spike changes shape just slightly. Your antibodies from last year's flu fit the lock mostly, but not perfectly. You still get sick, but it's mild. The second is antigenic shift. This is the nightmare scenario. Imagine a pig farm. Pigs can catch human flu, and they can catch bird flu. If a single pig cell is infected with a human virus and a bird virus at the same time, the genetic segments can shuffle like a deck of cards. The result is a Frankenstein virus. It might have the deadly interior of a bird flu, but the sticky H spikes of a human flu. When this new creation jumps to humans, no one on Earth has immunity. The antibodies do not recognize it at all. This is how pandemics are born. Let us leave the lungs now and follow the path of food. We descend into the digestive system. Here, we must make a critical distinction between two types of illness. We often group them together as food poisoning, but medically, they are very different. They are infection versus intoxication. The first category is infection. This occurs when you eat a living germ. The bacteria must survive the acid of your stomach, swim to the intestine, attach to the wall, and begin to grow. This takes time. The bacteria need to multiply to high enough numbers to cause trouble. Therefore, there is a delay. You eat the bad chicken at dinner, but you don't get sick until the next day, or even 2 days later. This delay is the incubation period. A classic example is Salmonella. These bacteria live in the intestinal tracts of birds and reptiles. If you eat undercooked chicken or raw eggs, the Salmonella enters your gut. They invade the cells lining the intestine. The body responds with fever and inflammation, gastroenteritis. The fever is the telltale sign that the bacteria are invading your tissue. The second category is intoxication. This is different. In this case, you are not sick because of the bacteria. You are sick because of a poison the bacteria made before you ate the food. Consider staphylococcal food poisoning. Imagine a chef with a small cut on his finger. He carries Staphylococcus aureus. He slices ham for a sandwich. The bacteria are transferred to the ham. The sandwich sits at room temperature for 5 hours. The bacteria grow. As they grow, they release a potent enterotoxin into the ham. Later, the sandwich is put in the fridge. The cold stops the growth. Or maybe the sandwich is cooked. The heat kills the bacteria. But, and this is the crucial point, the toxin is heat stable. The poison remains in the food even if the bacteria are dead. You eat the ham, you are swallowing a bag of poison. There is no incubation period for growth. The reaction is immediate. Within 1 to 4 hours, the body realizes it has been poisoned and triggers a violent ejection reflex. Severe vomiting and cramping ensue. But usually there is no fever, because there is no living infection attacking the tissue. It is a hit and run. We cannot speak of the stomach without mentioning the detective story of Helicobacter pylori. For decades, doctors believed that stomach ulcers were caused by stress, spicy food, or too much acid. They told patients to drink milk and relax. They were wrong. In the 1980 seconds, 2 Australian researchers discovered a spiral-shaped bacterium living in the stomach lining of ulcer patients. No one believed them. How could a bacterium survive in pH 2 acid? H. pylori has a trick. It produces an enzyme called urease. This enzyme converts urea in the stomach into ammonia. Ammonia is a strong base, it is alkaline. The bacterium surrounds itself in a bubble of ammonia, neutralizing the acid around it. It wears a chemical spacesuit. Safe in this bubble, it drills into the protective mucous lining of the stomach. The acid then burns the exposed tissue, creating the ulcer. One of the researchers, Barry Marshall, was so frustrated by the skepticism that he drank a beaker full of the bacteria. He gave himself gastritis to prove the point. He won the Nobel Prize. Today, we treat ulcers with antibiotics, not milk. This knowledge of the respiratory and digestive tracts dictates how we live our modern lives. It is the reason we cook our poultry to 165 degrees Fahrenheit to kill the salmonella infection. It is the reason we keep our potato salad on ice to prevent the staphylococcus intoxication. It is the reason we sneeze into our elbows to stop the aerosol transmission of the influenza virus. These systems are the most intimate connections we have with the planet. We take the world in and we let it out. Most of the time, the system works. The acid burns the invaders. The mucus traps them. The immune system patrols the borders. But sometimes, the waxy coat of the TB bacteria or the shifting spikes of the flu virus find a way through. As you rest, visualize the rhythm of these systems, the slow intake of sterile air, the quiet digestion of clean energy, the body filtering, sorting, and protecting. We have looked at the human body in depth, but the microbes do not need us. We are just one small habitat in a vast biosphere. In our next lesson, the penultimate chapter, we will leave the human host entirely. We will step out into the forest and the field. We will explore environmental microbiology. We will see how bacteria run the nitrogen cycle, clean our water, and recycle the dead to make way for the living. Lesson 19. Environmental Microbiology. For 18 lessons, we have focused our lens on the microcosm within. We have studied the wars fought in the bloodstream, the sieges of the cell wall, and the delicate balance of human health. It has been a story of us versus them. But this is a narrow view. If we stepped back and looked at the Earth from space, we would see that humans are merely recent guests on a planet owned and operated by microbes. Long before the first animals swam in the ocean or the first plant took root on land, bacteria were here. They engineered the atmosphere. They prepared the soil. They built the stage upon which all other life performs. In this lesson, we step outside the clinic and into the wild. We explore environmental microbiology. We will examine the biogeochemical cycles, the great recycling systems that move the elements of life from the air to the earth and back again. We will discover that without these invisible engineers, the gears of the planet would grind to a halt within days. Let us begin with the ground beneath your feet, the soil. To the naked eye, soil looks like inert dirt. But under the microscope, a single teaspoon of healthy soil is a bustling metropolis containing billions of bacteria, miles of fungal hyphae, and thousands of protozoa. This community is responsible for the carbon cycle. Visualize a forest. Trees grow, leaves fall, and animals die. If there were no microbes, this dead organic matter would simply pile up. The forest would be choked by its own debris. The carbon locked inside the dead wood would be trapped forever. But the microbes are the great recyclers. They are the saprobes. Bacteria and fungi secrete enzymes that break down the tough cellulose of the plant walls and the proteins of the dead animals. They release the carbon back into the atmosphere as carbon dioxide. This is not waste, it is breath. The plants breathe in this CO2 to build new leaves, continuing the circle of life. The decay of the old is the fuel for the new. But the most critical contribution of soil bacteria involves a different element, nitrogen. This is the story of the nitrogen cycle, and it is arguably the most important chemical process on Earth. Consider the air you are breathing right now. It is 78% nitrogen gas, N2. Nitrogen is the essential building block of life. You need it to build your DNA. You need it to build every single protein in your muscles and enzymes. Here is the paradox. You cannot use the nitrogen in the air. Neither can plants. The two atoms of nitrogen gas are held together by a triple bond, one of the strongest chemical bonds in nature. It is incredibly stable. Your body has no way to break it apart to get the raw atoms. You are starving for nitrogen while swimming in a sea of it. Who breaks the bond? Only bacteria. This process is called nitrogen fixation. There are specialized bacteria, such as the genus Rhizobium, that possess a unique enzyme called nitrogenase. This enzyme is a molecular hammer capable of shattering the triple bond. These bacteria take the inert gas from the air and convert it into ammonia, NH3. Ammonia is a form of nitrogen that life can use. The relationship between Rhizobium and plants is a masterpiece of evolution. These bacteria live in the roots of specific plants called legumes, plants like soybeans, peas, clover, and alfalfa. If you pull up a soybean plant and wash the roots, you will see tiny pinkish bumps. These are root nodules. Inside these nodules, the plant provides the bacteria with a safe home and plenty of sugar for energy. In exchange, the bacteria fix the nitrogen, turning the air into natural fertilizer directly inside the plant's roots. This is why farmers rotate their crops. One year they plant corn, which sucks nitrogen out of the soil. The next year they plant soy, which puts the nitrogen back in, thanks to the invisible microbial factory in its roots. But the cycle does not end there. Once the nitrogen is in the soil as ammonia, other teams of bacteria take over. Nitrifying bacteria convert the ammonia into nitrates. This is the form that most other plants, like wheat and oak trees, prefer to drink up. Finally, to prevent the soil from becoming toxic with too much nitrogen, denitrifying, bacteria step in. They take the nitrates and convert them back into nitrogen gas, releasing it into the atmosphere to complete the cycle. Fixation, nitrification, denitrification. It is a perfect, self-regulating wheel, turning silently in the dark earth, feeding the world. Now, let us move from the solid earth to the water. Water is the universal solvent, and because of this, it carries the waste of civilization. Every time you flush a toilet or drain a sink, you are sending organic waste carbon and nitrogen into the water supply. If we simply dumped this raw sewage into rivers, the bacteria in the river would feast on it. They would grow explosively. And as we learned in our metabolism lesson, growing bacteria consume oxygen. They would suck the oxygen out of the river, suffocating the fish and killing the ecosystem. This is called eutrophication. To prevent this, humans have built massive digestive systems made of concrete and steel. We call them wastewater treatment plants. But here is the secret. We don't clean the water. Microbes do. We just build the tank. Visualize the process of secondary treatment, often called the activated sludge system. The raw sewage enters a massive aeration tank. We pump huge amounts of oxygen into the tank, turning it into a bubbling cauldron. Inside this tank, we deliberately cultivate a zoo of specific bacteria and protozoa. We call this living mixture activated sludge. These microbes are hungry. We feed them the sewage. They feast on the organic matter, breaking down the feces and the food waste, converting it into harmless cell mass and carbon dioxide. They are the workers that scrub the water clean. After a few hours, we turn off the air bubbles. The heavy bacterial sludge settles to the bottom of the tank, leaving clear, clean water at the top to be released back into the river. Without these microbial janitors, our cities would be uninhabitable, buried under their own waste. We even use microbes to clean up our industrial mistakes. This field is called bioremediation. Imagine an oil spill in the ocean. The black crude coats the surface, killing birds and fish. Humans try to skim it off, but it is difficult. Nature has a better way. There are bacteria, such as Pseudomonas, that naturally eat oil. To them, petroleum is just another hydrocarbon, another food source. Scientists can spray fertilizer onto the oil slick to encourage these native bacteria to grow. As they bloom, they digest the oil, breaking the complex toxic chains into harmless byproducts. They literally eat the pollution. We are currently researching microbes that can eat plastic and fungi that can absorb radiation in the ruins of Chernobyl. This lesson changes our perspective. We are taught to fear germs. We scrub them from our hands. We poison them with antibiotics. We boil them in autoclaves. And yes, the pathogens are dangerous. But the pathogens are a tiny minority. They are the few criminals in a population of billions of peaceful citizens. The vast majority of the microbial world is benign, helpful, and essential. They are the breath of the soil. They are the filter of the water. They are the connection between the dead and the living. They are the only reason there is food on your table and air in your lungs. As you rest, visualize the planet not as a ball of rock and water, but as a living skin. Visualize the soil breathing. Visualize the roots trading sugar for nitrogen. Visualize the invisible engines keeping the biosphere running. You are not separate from this system. You are a part of it. The atoms in your body were once in the air, fixed by a bacterium, eaten by a plant, and consumed by you. Eventually, the microbes will reclaim those atoms to build something new. It is a comforting thought. Nothing is lost. Everything is transformed. In our final lesson, we will look at how humanity has learned to harness this transformative power. We will move from observation to application. We will explore applied and industrial microbiology. We will see how we use these tiny organisms to bake our bread, brew our beer, and manufacture the life-saving drugs of the future. Let the scale of the planet settle in your mind. The cycle of the carbon, the bond of the nitrogen, the cleaning of the water, the eternal return. Lesson 20. Applied and Industrial Microbiology. We have arrived at the end of our curriculum. Together, we have journeyed through the history of life. We have peered through the lenses of the first microscopes. We have mapped the architecture of the cell. We have studied the ancient wars between the immune system and the pathogen. We have stood in the soil to watch the cycles of the earth. Throughout all these lessons, our role has been that of the observer. We have watched microbes do what they have done for billions of years. We have studied them as forces of nature, sometimes fearing them, sometimes admiring them. But in this final chapter, the relationship changes. We stop being observers and become architects. We stop fighting the microbes and start employing them. This is the world of applied and industrial microbiology, often called biotechnology. Biotechnology is a word that sounds futuristic, summoning images of genetic splicing and glowing test tubes, but in reality, it is as old as civilization itself. The first biotechnologists were not scientists in white coats. They were bakers and brewers in ancient Egypt and Mesopotamia. Consider the loaf of bread. This is a product of microbial engineering. Thousands of years ago, humans discovered that if you left dough to sit, it would rise. They didn't know why. They didn't know that a unicellular fungus called Saccharomyces cerevisiae yeast was feasting on the sugars in the flour, performing fermentation, and releasing carbon dioxide bubbles to inflate the dough. Consider the glass of wine. They didn't know that the same yeast was turning grape juice into ethanol, creating a beverage that was often safer to drink than the water. Consider the wheel of cheese or the jar of yogurt. They didn't know that lactobacillus bacteria were converting milk sugar into lactic acid, preserving the protein and changing the flavor. These were the classical biotechnologies. They relied on the natural behavior of microbes found in the wild. We simply provided the food and the warmth, and the microbes did the work. But today, we have moved from the kitchen to the factory. Industrial microbiology takes these ancient processes and scales them up to a magnitude that is hard to comprehend. Visualize a modern industrial facility. Rows of gleaming, stainless steel tanks tower towards the ceiling, 2 stories high. These are bioreactors, or fermenters. Inside each tank is a liquid medium, swirling with nutrients. And swimming in that liquid are trillions of microbes. The bioreactor is a life support machine. Sensors monitor every variable: temperature, pH, oxygen levels, stirring speed second by second. Computers adjust the inputs to keep the microbes in their perfect Goldilocks zone for maximum efficiency. What are they making in there? They might be making citric acid for your soft drinks. Almost all the citric acid in the world is not squeezed from lemons, but produced by a mold called Aspergillus niger, growing in a tank. They might be making enzymes. The stain-fighting power in your laundry detergent is actually a collection of bacterial enzymes, proteases and lipases, harvested to digest the food stains on your clothes. They might be making amino acids to supplement animal feed, or vitamins like B12, which are difficult to synthesize chemically but easy for bacteria to produce. But the true revolution of modern microbiology is not just growing microbes, it is reprogramming them. This is the era of genetic engineering, or recombinant DNA technology. For billions of years, a bacterium could only do what its own DNA told it to do. It was limited by its own evolution. But in the 1970 seconds, scientists discovered that DNA is a universal language. The code ATCG is the same in a human, a fish, and a bacterium. This meant that, theoretically, if you took a gene from a human and put it into a bacterium, the bacterium could read it. To do this, we developed molecular tools. We found restriction enzymes. These are molecular scissors that cut DNA at specific points. We found ligase. This is molecular glue that pastes DNA strands together. We found vectors. These are the delivery trucks, usually the plasmids we discussed in Lesson 6, that can carry a new gene into a bacterial cell. The most triumphant example of this technology is the story of insulin. Consider the diabetic patient. For decades, they survived by injecting insulin harvested from the pancreases of slaughtered pigs and cows. It worked, but it wasn't perfect. It was animal protein. It caused allergic reactions. It was expensive and required millions of animals. Then came the idea, why not teach a bacterium to make human insulin? Scientists took the human gene for insulin, the instruction manual found in your own DNA. They cut it out. They pasted it into a plasmid. They inserted that plasmid into a common E. coli bacterium. The bacterium read the new code. It didn't know it was human code. It just followed instructions. It began to churn out pure, perfect human insulin. We grew these bacteria in massive bioreactors. They became microscopic pharmaceutical factories. Today, every diabetic who injects insulin is using a product made by genetically modified bacteria. It is safer, cheaper, and limitless. We did the same for human growth hormone and clotting factors for hemophiliacs. And now we stand on the brink of the next frontier. We have moved beyond just cutting and pasting genes. We are learning to edit them. We have discovered a system called CRISPR. Ironically, CRISPR was discovered as an immune system for bacteria, a way for them to remember and cut up attacking viruses. We have repurposed this bacterial shield into a tool that allows us to edit the genetic code of any living thing with the precision of a word processor. We are looking at a future where we might use modified viruses to deliver cures for genetic diseases directly into human cells. We are looking at bacteria engineered to eat plastic waste in the oceans or produce biofuels to replace gasoline. We have come a long way from the animalcules of Leeuwenhoek. We started by looking through a piece of glass at a blurry speck. We ended by rewriting the code of life to save our own. This concludes our course. Let us take one final moment to synthesize what you have absorbed. Over 20 lessons, you have built a mental map of the invisible world. You know the structure of the prokaryote and the eukaryote. You know the shapes of the cocci, the bacilli, and the spirals. You understand the exponential math of growth and the fiery chemistry of metabolism. You have seen the DNA scroll and the viral hijacker. You have explored the fungi of the soil and the parasites of the water. You have armed yourself with the knowledge of sterilization, antibiotics, and vaccines. You have toured the fortress of the human body, from the acid of the stomach to the barrier of the brain. You have seen the cycles of the planet and the factories of industry. The invisible world is no longer a realm of mystery or fear. It is a realm of complexity, beauty, and order. You understand now that you are not an individual entity living in an environment. You are an environment. You are a host. You are a colony. You are a partner in the vast, interconnected web of life that covers this planet. As you drift into the final moments of wakefulness, let this perspective bring you peace. The world is working exactly as it should. The bacteria are cycling the nitrogen. The algae are making the oxygen. Your immune system is watching the gates. Your cells are humming with energy. You are part of the system. You are safe. The class is dismissed. The journey is complete.